Hello. Noise for some reason.
Will have fruitful, rewarding exchange of knowledge of the concerned topics of the conference in next 2 days. I wish you every success with this important conference, and I look forward to learn about the outcome with your valuable suggestions and feedbacks. We all know that at the beginning of any knowledgeable event, we all take blessings from the Almighty. Now, I would like to start our conference with Ma Saraswati Vandam. Thank you.
Good morning, our respected chief guest, Dr. Shailendra Sraf sir, guest of honor Sri Atul Nasa sir, respected director sir, Director General NIT, Dean ma'am, and all dear participants. Now, I would like to invite Director Pharmacy, Dr. Abhijit Majumdar sir, to enlighten us all about the conference. Thank you.
A very good morning to all our beloved participants. Respected Dr. Shailendra Sraf sir, Vice President Pharmacy Council of India, Sri Atul Nasa sir, Deputy Drug Controller Delhi, Professor Praveen Suneja sir, Director General of Noida Institute of Engineering and Technology, Greater Noida, Dr. Vinod M. Kapse sir, Director NIT, Dr. Rupa Majumdar, co-convener of this conference, all directors of various pharmacy institutions of the country, head dean directors of our institute, all respected dignitaries and beloved delegates. At the very onset, I would like to extend a very, very warm welcome to Dr. Shailendra Sraf, Vice President of Pharmacy Council of India, who has taken his very valuable time amidst his very busy schedule to address our AICT-sponsored conference titled Antibiotic Resistance, Causes and Future Challenges in Digital India. Dr. Sraf has taken a leading role in the upliftment of our profession by using the platform of Pharmacy Council of India. I would like to extend a very hearty welcome to Deputy Drug Controller Government of Delhi, Sri Atul Nasa sir, who also has taken a very key step in maintaining the regulatory guidelines of the profession of pharmacy. I am welcoming Professor Praveen Suneja sir, Director General Institute, and Dr. Vinod N. Kapse, Director of the Engineering Institute, in the inaugural program of this conference. This year, though the country has been suffering due to the corona infection, but believe me that this year has been a landmark in the history of our NIT Pharmacy Institute, because in this year only we had been selected the 49th rank among all pharmacy institutes of the country as per NIRF ranking, after being selected for the 3 consecutive years, NIRF 2018, 2019 and 2020. In the same year 2020, we have been re-accredited by National Board of Accreditation or NBA for our B-Farm program. And this year, we are fortunate enough to receive 3 research promotion schemes, 2 from government of, 2 from AICTE New Delhi, and 1 from our affiliating university of amounting to 45 lakhs. So 45 lakhs research grant has been received by our institute in 1 year only in 2020. In fact, I am very fortunate to announce that All India Council of Technical Education New Delhi has been kind enough to sponsor this national e-conference on this antibiotic resistance causes and future challenges of the country. As we all know that antibiotics were considered as magic bullets since the discovery of penicillin in 1929 by Sir Alexander Fleming. But unfortunately, these antimicrobial agents are gradually losing its efficacy to control various infections. Infections including pneumonia, tuberculosis, and gonorrhea, they are becoming harder and sometimes impossible to tackle when these antibiotics are becoming ineffective. Now, there are certain factors which are responsible for this development of drug resistance. The random use of antibiotics coupled with the ignorance of the patient as well as this high cost of these medicines has led to the patient not to complete the dosage regimen. Now, whenever the patients are not completing the dosage regimen, these chemicals are acting as a mutating agent due to which the bacteria, the microbes are developing resistance for that particular agent. Now, self-medication, ignorance, and non-completion of proper dosage regimen has led to a great problem which we are addressing today as multi-drug resistance of the microbial strains. There are other factors too. We are using these antibiotics at random for animals too, so that whenever we are drinking their milk, we are unnecessarily consuming various chemicals. Antibiotics are subsidiary agent for use for the controlling of various infections in agricultural sector also. Therefore, this has led to a very high economic bioburden in the healthcare system. Now, when this problem arises, it increases the hospitalization period of patients who are suffering from infection. Now, moment this duration increases, so automatically this is increasing the treatment cost. Therefore, the public health leaders should establish a pan-surveillance system coordinated at national and international levels, an ongoing analysis and a mandatory reporting system of the antibiotic resistance needs to be developed. It is the need of the hour. Both domestic and global policies need to be conventional and adhered to overcome the misuse of drugs and antibiotics nowadays. In fact, today we have organized this seminar to discuss in detail about the various causes that has led to this global menace. I am really thankful to All India Council for Technical Education, New Delhi, as well as the management of our institute for being kind enough to sponsor this seminar. In fact, this seminar will provide an unique opportunity to the faculty members, experts from academia, various experts from hospitals and industries, so that they can have a joint platform to attend some brainstorming session and then find out some key strategies which may be forwarded to the government to control this drug resistance. I am happy to share with you that we are having oral and poster sessions also, where a number of participants will be presenting their views by oral and poster presentation, so that I hope that with these 3 years, this 3 days conference will be very fruitful for the faculties, for the academicians, for the people from hospitals, as well as from pharma industries. Thank you very much.
Thank you, sir, for your valuable speech, and we are really blessed with your kind words. Now I would like to welcome Director NIT, Dr. Vinod M. Kapse sir, to bless us with his enthusiastic words for our conference. Sir, please.
Good morning to all. Chief guest of today's session, Dr. Shailen Saraf, Vice President, PCI New Delhi. Guest of honor, Shri Atul Kumar Nasa, Deputy Drug Controller Department. Professor Praveen Soneja, Director General, Director Pharmacy, Dr. Rupa Majumdar, Dean Pharmacy, Professor Salauddin, all the directors from various college, participants, and faculty members. First of all, on behalf of NIT Group, I would like to welcome all the dignitaries for this AICT sponsored conference. Also, I would like to congratulate the Institute of Pharmacy for organizing this AICT conference. Definitely, this conference will provide a platform where the different researchers contribute their research work. Also, during this pandemic, the two people played an important role, like one is doctor and second one is pharmacist. So I would like to salute for their remarkable contribution during this pandemic. Definitely they played a very vital role. So salute to them. Also, I would like to congratulate the Institute of Pharmacy for achieving rank in NIRF ranking under 50 under the able leadership of Dr. Majumdar and Rupa Majumdar. So they are playing a vital role. Definitely their contribution is remarkable. So once again, this conference will play an important role for presenting their research work. Also, the various eminent speakers from the country will give important input, valuable input to all the young researchers, and they will learn a lot from this conference. Thank you.
Good morning, all. Can you hear me?
Yes, sir.
Okay. Good morning, all. At the outset, let me thank AICTE for choosing such a relevant subject which affects all of us and society at large. And thank you, Shri Shailen Saraf, Chief Guest for the function, and Shri Nasa for accepting our invitation. On behalf of entire NIT leadership team, I thank you for sparing your valuable time and gracing this occasion. I also thank the Dr. Majumdar, Dr. Rupa Majumdar, and entire team of pharmacy for organizing this event. Thanks all the distinguished guests for being part of this conference and enlightening us over next 2 days. Thank you all the dignitaries, speakers, and I wish everybody's best. And I'm sure this will come out with remarkable findings which will help our society at large. Thank you all.
Thank you, sir, for your kind words, which are really have a great blessing for the conference. Now, I would like to welcome our guest of honor, Shri Atul Kumar Nasa, sir, to bless us with his knowledgeous words based upon his vast experience to start the conference. We are really grateful to have such an esteemed person who is working as a Deputy Drug Controller, Drug Control Office, Delhi. Dear participants, you all will be glad to know that he has awarded as the Best Drug Control Officer Award in 2017. He has also been accorded the Fellowship of Indian Pharmaceutical Association, considering his professional trajectory in the field of pharmacy and drug regulations. He is a member of Drug Consultative Committee and also an active member of the Pharmacy Council of India. An officer with more than 25 years of experience in enforcement of drug laws, Sri Atul Kumar Nasa sir has participated in various national and international conferences on pharmacy and related fields. He has been a part of all programs organized by WHO, CDS, CO, and other industry associations on myriad topics related to the drug regulations. His leadership qualities, cooperative attitude, admirable acumen in all the sectors of pharmacy, and the spirit of undying professional fraternity are always recognized. It's my immense pleasure to welcome you, sir, for your kind words. Sir, please. Sir will be joining soon. Please wait for some time.
क्या कहेंगे पापा इन्होंने? किस बारे में बात करें?
NASA sir will be joined us soon. Now, I would like to welcome our chief guest of the day and Vice President of Pharmacy Council, Dr. Shailendra Saraf sir. Professor Shailendra Saraf is a well-known pharmacy professional actively involved in promoting pharmacy education and research. He has served as Vice Chancellor of Hemchand Yadav University, Durg, Chhattisgarh. He is an active member of All India Council of Technical Education, New Delhi, as well as National Board of Accreditation and National Assessment and Accreditation Council. Professor Saraf is associated in different capacities with central statutory regulatory accreditation bodies of higher education, that is Drug Technical Advisory Board, Pharmacy Council of India, AICTE, NBA, NAC, DST, and UGC. He has been awarded with many national and international awards of repute. Professor Saraf is actively involved in research activities for last 3 decades and working in the field of herbal drug technology and medicinal plant research. He has successfully completed several research projects supported by extramural research funding agencies like UGC, AICTE, MP Cost, CG Cost, DST, etc. He was instrumental in establishing and implementing several institutional projects like UGC SAP, AICTE MODROC, DST FIST, and DST sponsored National Center for Natural Resources. Professor Saraf, in his long and wide academic career spanning over 3 decades, has supervised 15 candidates for their PhD degree. He has contributed more than 200 national and international publications in different eminent journals of national and international repute. He has authored 4 books, chapters, and filed 10 patents. Now I would like and welcome Sir. Sir, please. Sir, you are not audible. Sir?
Now I'm audible?
Yes, sir.
Okay, thank you.
Yes, sir. Thank you, sir.
Oh, thank you very much, madam, for nice words. It's human weakness to listen to good words for himself. So I'm also victim of that. So thank you very much for nice words. And on board, the director of NEIT, Dr. Kapse, my friend Dr. Majumdar, Dr. Rupa Majumdar, Dr. Nasa, Dr. Vijay Bhalla, and other leadership of Noida Institute. It's really it's my proud privilege and a definite pleasure to be here amidst all of you. And I seize this opportunity to place on record my thanks for inviting me for such a prestigious seminar. It's better if you ask to put on mute, otherwise it's difficult for me to speak. So I am really thankful to be here on this platform, which is organized by Noida Institute of a group of institutions. And I can see in the backdrop of Dr. Majumdar, it is reflected. This is an IRF ranked within 50 and MAC assessed institution. It speaks on its own, though it's a 15-year-old, but it's providing leadership in the field of technical education in general and pharmacy education in particular. So my good wishes and keep doing good for the profession. So I know I was supposed to speak after Dr. Nasa, but he is a frontline warrior and I can understand he could not attend on time. So he's always excused because he's serving the nation and profession. So my good wishes and good morning, Dr. Nasa. And I brought greeting to all of you this morning. And I greet all of you on behalf of Pharmacy Council of India on my own behalf. And I would like to thank Dr. Majumdar for selecting such a important topic because during this last 6-7 months we are fed up with this corona and COVID. So there are clear-cut indication of this information over. There are two distinct groups in the society. One is suffering from the overload of information, and another is confusion, overload of confusion, because most of the informations are available in English and Hindi. Local languages are not available any information. So there are two type of groups, and importantly, 100% people are victim of fear psychosis, and that is the point where we have to switch our attention towards some other things also, so then we can live psychologically better, we can lead a better life. So this initiative, you are in this pandemic, you are taking leadership of time, you are organizing the activity. At the same time, you are selecting it a topic of very important, very importance worldwide. So when we talk about antibiotic resistance, so it recalled me just the interview given by Fleming in 1945 when he was awarded with the Nobel Prize, and he was talking to media and the experts, and he said we should be careful as far as the use of antibiotic is concerned. So he warned about the society. But this is human nature. We never respond to the this type of warning until we face or we counter with all such problem. So now we are in a typical condition, but nobody is still realizing what is the problem, because the most important observation is there is no work going on new antibiotic development, though there are different factors. And after 1968, only two systematic for antibiotics were developed. And after that, there is even with the I checked with the US FDA, there is no systemic new antibiotics are listed in, and we can expect in near future. It means if we are because we are blessed with the two most important inventions in the last century, were antibiotics and this vaccines. And one, we easily forget the problem of when we are not having the antibiotics, and one-time single infection taking the lives of a million or billion people. And antibiotics proved to be a boom at that time, but slowly we forget the importance of antibiotic. And it's not we can say alarming, but it is already we have reached to this level where we are approaching towards pre-antibiotic age again. Why I am saying so? Because out of this 2 million people every year in US alone, 70% are resistant to, and one of the at least one antibiotic for resistance, facing resistance. And if you talk about the survey carried out the UK government, so this is going to affect the economy of $100 trillion. So it is going to affect everyone. And what is the solution for that? Simple saying the antibiotic is developing the resistance. This is a simple, we can say, mechanism also. And the real problem exists because we are not having any approach for the ideation or for the forecasting of such type of things. In any branch of the science, if you see, even in a profession, there are thought leaders who are time to time forecasting something, and they are giving the guidelines also. Even if you see the example of this pandemic, nobody predicted. There was no warning. There was no alarm for any corner. It means there is ideation in this particular field is 100% missing. So we have to think about the ideation. We have to think about the forecasting. Then only we will be able to give the solution in the pharmacy profession. We are patting our back, no doubt. We are producing the drugs, but this is not sufficient. We are not really looking after the 360 view of our profession, and that is expected, because when we are patting our back for the supply of the drugs for the world, the real heroes wear 3-layer mask and simple, we can say, sanitizers, and they are not coming under the purview of pharmacy practice and all that. We are not giving due importance. So we have to, this is time to sit down for some time, churn on certain issues, and come out with a 100% 360 policy, so that we can cater the need in a significant manner. one important thing we have, this is the blessings in disguise. The world has recognized the existence of pharmacists. We were fighting for our recognition, but this particular pandemic time gave us opportunity, and we are recognized, we are commanding respect as well. Why I am saying so? Because across the globe, there was not even a single case reported where pharmacists are found in unethical practices. During this pandemic, this total supply chain was not disrupted. During this pandemic, no drugstore was closed. During this pandemic, no pharmacist denied the services or practices for the patient, for the well-being of the patients. So this is the contribution of all the pharmacists working in different facets of the pharmacy profession to bring the laurels and bring the recognition that this breed is also existing in the society. When we talk about antimicrobial resistance or antibiotic resistance, so who is the better agent to work in a systematic manner? Though there is a full-fledged advisory issued by the government of India, DCGI of the country in December 2019, and it talks about the prevention of this sale of antibiotics under prescription. That is already categorized under Schedule H, I believe, H and H1. But still this is supplied. Whenever we talk about the misuse of antibiotic, so we can sure that this drug has been given by pharmacists. So if the pharmacists of the country or the world are playing their role, professional role, then we can minimize the problem of antibiotic resistance. So this is the high time to just to remember the pharmacist oath and work according to the profession. This is not anything over and above your expected from your duties. This is the duty of your, because you have to work as per the Drug and Cosmetic Act and Pharmacy Act. And this is mentioned, you cannot sell the drugs without prescription, but that is not the true picture. And as far as the research is concerned, we have to seriously think and we have to form a group so that we can lead this particular crisis through academia. Because the industry always think about the equation of loss and profit. And as far as the invention of this antibiotics concerned, it requires lot of money. It requires really pooling of money, and that is next to impossible for the individual companies to come out with new drug molecules. Why? Because if anybody is coming out with a new systemic drug, the most important and tedious problem is this is the clinical trials. And if you are claiming about any bacteria, so you have to test your antibiotic against each and every microbe, and getting the volunteers, identification of volunteers suffering from those specific bacteria is a big problem. And when we talk about the economy of antibiotic, it's really on the back front, because the only statins like drug which are not life-saving drug, but they are commanding the respect as far as the volume of the money of the manufacturers concerned. So antibiotics normally the active dose are required for 5 to 10 days. But if you talking about lifestyle drugs like sugar and blood pressure and cholesterol lowering drugs, they are supposed to take months together. So they bring better profit to the industry. So the industry people are not involved in such type of inventions, and this is a high time to discuss and to give some idea to the regulators about this drug development process. Because even if this is pandemic, we have to understand, we have to assess the risk versus profit concept as far as the new drug development is concerned, whether it is a vaccine or it's an antibiotic. If you are not thinking in this direction, we can say that we are going to lose our share in the respect in the medical profession. And while are we talking about the antibiotic resistance and antimicrobial resistance, so we have to understand the concept of microbiome also. Because the main problem with the development of antibiotic is we have to take care of 3 billion microbes existing in our body, and total weight of this particular microbe available in our gut is more than 2 kg. So this is if we want an agent which is targeting the enemies, But at the same time, this is familiar. This is symbiosis with the microbes available in the gut. So we have to find out certain out-of-the-box ideas, and here he is mentioned in this topic itself covers the antibiotic registers in the time of electronic, we can say, era, and this is time when the real drug development is proposed based on the this kind of phenotypic observations among the patients with the help of big data analysis and integration of artificial intelligence. So this is a well-taken topic. We have to think about the out-of-the-box idea as far as new drug development is concerned. And believe me, anywhere in the world, such type of non-profitable drug development can happen only with the support of academia. And I believe this particular conference must be a milestone as far as the beginning of new era is concerned. So I hope the 3 days discussions, I have seen the list of experts. All experts are coming from different domain of the pharmacy profession. They will come out, and certainly these organizers will prepare a report so that the regulators will think about on such issues. So I am thankful for Dr. Mazumdar and institution for inviting me and giving me an opportunity to interact with all of you and to see the face-to-face my good friends Dr. Bhalla and Dr. Nasaji. And it's really my pleasure to be here and looking forward to have the good discussions, fruitful deliberations, and a document, a policy document coming out of this discussion is prepared and submitted to different regulatory bodies and statutory bodies. So I wish the organizers to carry on carry out your leadership role in the field of academia in this tough time, and at the same time thankful to the organizers for giving us opportunity to connect all the pharmacists of this country. Thank you very much. All good wishes. God bless all of us, and be safe and be healthy. Thank you very much.
Thank you, sir. Thank you a lot for your valuable talk in regard to the conference. We are really blessed with your presence today, sir. Sir, definitely your suggestions will help us to take a leading role to control this global problem. After various deliberates by esteemed speakers from industries, hospital, academia, we will try to frame a strategic guideline which will forward in due course to PCI as well as to government regulatory bodies for necessary action. Thank you, sir.
Thank you. Thank you.
Now, I would like to welcome and invite our guest of honor, Shri Atul Kumar Nasar sir, to bless us with his knowledgeous words based on his vast experience to start the conference. As we already know, Sir has awarded as the Best Drug Control Officer Award in 2017. He is a member of Drug Consultative Committee and also an active member of Pharmacy Council. His leadership qualities, cooperative attitude, admirable acumen in all the sectors of pharmacy, and the spirit of undying professional fraternity are always recognized. It's my immense pleasure to welcome you, sir, for your kind words. Sir, please.
Am I audible?
Yes, sir.
Thank you.
Thank you, sir.
Good morning, everyone. My special thanks to Dr. Shraf. Already he has covered most of the points on antimicrobial resistance. I also welcome Dr. Bhalla, and my sincere thanks to Dr. Salahuddin and Dr. Majumdar from the NIT Institute for organizing this 3 days workshop or a national conference on antibiotic resistance, cause and future challenges in digital India. This is a very well said topic taken, like Dr. Shraf has told, because from last 6 to 7 months, we are only discussing COVID and COVID related issues. But at this time, once we are total stabilized and moving forward. So this topic of antimicrobial resistance is well chosen, and I will share some points here, being a regulator working in drug control department. If you see the antimicrobial resistance, there was a news in the media long back that around 50% of the population in our country is suffering from antimicrobial resistance. So this data was very shocking. 50% of population suffering from antimicrobial resistance, antibiotics are not affecting, and a big challenge to the entire country, to the entire community, to the entire regulations, and to the entire pharma industry. In 2017, in the April 2017, the Ministry of Health and Family Welfare, Government of India, along with the Office of DCGI, that is CDACO, they developed a national action program on antimicrobial resistance. So once this challenge was there, so this national action program was being formulated by Government of India, and Government of India is working from time to time to strengthen and to make different advisories under this program to stop antimicrobial resistance. In 2013, now if you see Drugs and Cosmetics Act, earlier the antibiotics, all the antibiotics were included on the category of Schedule Schedule H drugs, that is prescription drugs. But later on, once the government was seeing that the antibiotics are widely misused, antimicrobial resistance is on rise, on 30th of August 2013, Government of India brought a notification that some of the latest generation antibiotics, not all the antibiotics, latest generation antibiotics will be covered under the definition of Schedule H1. And in Schedule H1, if you see the three categories, one is the habit-forming drugs, second is the anti-tuberculosis drugs, and third category is the latest generation antibiotics. So these three categories of drugs were notified under Schedule H1 under the notification dated 30th August 2013, and which is being governed by Rule 65 and Rule 97 of the Drugs and Cosmetics Act and rules made thereunder. Now, under Schedule H1, what was the necessity of bringing the Schedule H1? In each and every pharmacy, each and every chemist have to maintain a proper register for selling of these medicines which are categorized under Schedule H1, like latest generation antibiotics, anti-tuberculosis drugs, and habit-forming drugs. And chemists have to maintain a proper register prescribing the name and address of the patient, name and address of the prescriber or a doctor, quantity sold, and balance in hand, so that regulatory bodies or the drug control officers can visit any shop checking the records of the Schedule H1 medicines. Here I add, why habit-forming drugs? Because drug addiction was on rise. So once the drug addiction was on rise, drug abuse was on rise, these hypnotics and sedatives or habit-forming drugs ought to be controlled. That's why they are categorized in Schedule H1. In tuberculosis drugs, that is anti-TB medicines, patients were not taking the entire course of the anti-tuberculosis medicines, or they were getting these medicines without the proper guidance of a doctor. That's why anti-tuberculosis drugs are also kept in Schedule H1 drugs. And then is the latest generation antibiotics, because antimicrobial resistance was on rise. Even I tell you, during the COVID time, when ICMR and WHO has given a statement that HCQS tablets can be used for the prophylactic treatment of COVID, people started taking HCQS tablet of their own without the prescription of a doctor, without the guidance of a doctor, and there was acute shortage of HCQS tablet. HCQS tablet was earlier under the category of Schedule H. Later on, seeing the conditions, seeing the misuse of the HCQS tablet, government brought this medicine under the category of Schedule H1. So one more medicine was added in Schedule H1, that was the hydroxychloroquine tablet during the COVID time. So this how system is going on, categorizing various medication under the category of Schedule H1, so that their sale and their proper distribution is to be controlled. Now, question comes: what are the causes of antimicrobial resistance? Why it is on rise? Why WHO is saying that there are a number of patients who are suffering from antimicrobial resistance? The very first reason for antimicrobial resistance is misuse, overuse, or inadequate use of antibiotics. Sometimes doctors are prescribing the antibiotics even though it is not required. So this misuse or overuse or inadequate use of antibiotics is to be stopped. Second point is, when these antibiotics are sold without medical supervision, or you can say without the prescription of a registered medical practitioner, automatically antimicrobial resistance is on rise. Each and every chemist, each and every pharmacist has to play an important role, not to sell these schedule H1 or H drugs without the prescription of a registered medical practitioner. This is an important part. Then one of the reasons is prophylactic use of the antibiotics before surgery. Before surgery, if we are taking the antibiotics for prophylactic use, it may be one of the cause for antimicrobial resistance. Antibiotics used in the viral infection, this is very important. I take an example here. If we have got a viral fever, now virus has got its own life from 3 to 5 days. So there is no purpose of taking antibiotics, particularly when you are having the viral fever. So if you are starting taking the antibiotics in the viral fever, which is not affecting at all, which is having no use in the viral infection, we have to stop misuse of antibiotics. Another reason is spread of resistant microbes in hospitals. Now, due to unhygienic conditions, if the microbes are being developed in the hospital premises, this may be one of the cause for antimicrobial resistance. So each and every hospital, each and every institution where patients are being admitted, they have to take utmost care, particularly maintaining all hygiene conditions. This is very important. Then, incomplete course of the treatment taken by the patient. Here I quote an example of anti-tuberculosis medicines. We have got DOT centers all over the country. So if the tuberculosis patient stops his medication in between, or he is not taking the complete medication, tuberculosis will not be treated. Similarly, for antimicrobial resistance also, this is one of the cause. If we have to take the course of antibiotic for 3 days or for 5 days as being prescribed by a doctor or a registered medical practitioner, depending upon the body illness, we have to complete the course. If we stop the treatment in between, this may be one of the cause of antimicrobial resistance. Then antibiotics in animal feed. This is a big challenge. We are taking, there is one discussion on, disposal of unused medicines, disposal of expired drugs. Sometimes we people throw the unused medicines or expired drugs in dustbin, particularly antibiotics. And if these antibiotics, which are, thrown in the dustbin and they are later on, they are taken by the animals, and we are taking that animal feed, or we are taking that all, from poultry eatables, we are getting the, antimicrobial resistance. And then very big challenge is self-medication. We have to stop self-medication. This is required. Because if we are taking any medicine, not only antibiotics, I can say any medicines which is categorized under Schedule H or Schedule H1 without the guidance of a doctor, without the prescription of a doctor, and we are starting taking antibiotics of our own, thinking that we will be cured from this disease. This is one of the major cause for antibiotic or antimicrobial resistance. Dr. Shroff has told pharmacy, yeah, we are best in the world. Our country is exporting lot of medicines all over the world. 60% of our formulations are exported. And rightly said, our Honorable Prime Minister, vocal for local. This is the right time. This COVID has given us a lesson that we have to become self-dependent, Atam-nirbhar. If our pharmaceutical industry is well strong, we should not be dependent on the other countries. And big challenge was once this COVID started. we were getting most of the APIs from China. But due to this COVID and due to the China issues, we were not getting the active pharmaceutical ingredients from the China, and then there was acute shortage of formulation in the country. So with the advice of our Prime Minister and with the help of the support of pharmaceutical industry, all API pharmaceutical industry all over the country, they have made their system how to go forward. Let us not depend upon China or other countries. We are the professionals, we are the pharmacists. We can manufacture medicine, we can manufacture APIs, we can manufacture each and everything. So this is the right time. We have to understand that India is the pharmacy of the world, what Modi ji has told, what everybody says. And if India is the pharmacy of the world, we can be the best conditionally. We resolve all the issues related to our profession, related to pharmacists, related to Drug and Cosmetic Act, related to regulations, and related to all other points. Here I tell, as a pharmacy professionals, what role we have to play. This is important. Now we have to ensure quality drugs to the consumers, including antibiotics. If the antibiotics given to the consumers are not of quality drug or not of quality medicines, they are NHQ drugs, not of standard quality drugs, their action will be less, and automatically, chances for AMR, antimicrobial resistance, will be increased. So pharmacist has to play an important role by providing the quality medicines to the consumer, though it is the moral duty of a drug control department or a regulatory department to ensure safe, efficacious, and quality medicines to the consumer. But on the other side also, pharmacist has to play an important role. Pharmacists have to sell all these medicines. Recently, again, I am repeating, pharmacists have to sell all these medicines only on the prescription of a registered medical practitioner. Don't dispense any Schedule H or H1 drug to any of the consumer until and unless he or she is carrying valid prescription of a registered medical practitioner. Pharmacist has to play an important role by avoiding multiple dispensing of a prescription. Now, if a single prescription is being dispensed, this is well said under Rule 65 of the Drugs and Cosmetic Act. I'm sorry to say, we are not, implementing even till date. Most of the country regulatory department are not implementing. It is well said in the Rule 65 that once a prescription is to be dispensed, it is to be stamped that these medicines are already distributed, so that prescription should not be misused by the other counter, by going to the other counter and getting the repetitive medicines, particularly habit-forming drugs. There should not be any substitution. Now, whatever the medicine is being prescribed by a registered medical practitioner or a doctor for any patient, for any disease, we have to dispense the actual same medicine which is being prescribed. Substitution is not allowed, and it is a violation under Rule 65 of Drug and Cosmetic Act. Maintenance of proper records of Schedule H1 and Schedule H medicines. This is why I have told in the beginning also that each and every pharmacy has to maintain records of Schedule H1 drugs. And most important part is storage conditions. I have seen during our visits, sending my officers, most of the chemist shops are keeping the antibiotics in the refrigerator. Antibiotics are not to be stored in the refrigerator, where it is written, labeling condition to be seen, it is written on the label of an antibiotic that it is to be stored in a cool place, not in a cold condition. So in a refrigerator, the temperature is 2 to 8 degrees centigrade. So until and unless it is not specified, you don't store any antibiotic in the refrigerator and store the antibiotic at the required temperature as mentioned on the label. So with these my points, being a regulator, I can simply say we can stop antimicrobial resistance. A regulator has to play an important role. A pharmacist has to play an important role. Pharma industry has to play an important role. And each of us, we can join hands. Pharmacist is a carbon-carbon double bond between a physician and a patient. He is playing a very major role. So once a prescription is being prescribed by a doctor, it is to be dispatched by a pharmacist, it is to be given to the patient. So while dispensing, prescription by a pharmacist has to take utmost care, seeing what are the regulations, what are the laws, and what he has to do, what he has not to do. With these remarks, my special congratulations to NIT Institute, and I can simply say that your 3-day celebration, like what Dr. Shroff has told, you please come up with an outcome, concrete outcome of this conference, these seminars, considering the views of various speakers, various spokesmen, and these recommendations of your conference or seminars can be forwarded to Government of India, Ministry of Health and Family Welfare, even to Pharmacy Council of India for implementation and for their kind consideration. So with these remarks, I can simply say this is a COVID time, and I request each and everyone to please stay safe, take care. Thank you very much. Namaste. Samir.
Now, I would like to Dr. Sanchita Das, ma'am, to propose the vote of thanks. Ma'am, please.
A very good morning to Chief Guest of Honor, to Director General Sir, Director Sir, NIT, the Director, Pharmacy Institute, the Dean Ma'am, Pharmacy Institute. We all know that antibiotic resistance is one of the biggest threat to global health. And I hope all the participants and all the viewers have gained a brief knowledge and valuable knowledge in this regard of this ceremony, in this inaugural ceremony of e-conference. On behalf of NIT Pharmacy Institute and all the entire organizing committee, as well as my own personal, I, Dr. Sanjita Das, HOD Pharmacology, would like to extend our sincere thanks to all the the chairman, All India Council of Technical Education, Dr. Anil D. Sahasrabuddhe, for sponsoring National E-Conference on Antibiotic Resistance, Causes and Future Challenges in Digital India. I would like to take the opportunity to extend our thanks to the NIT management, Dr. Sarojini Agarwal ma'am, chairman, and Dr. O.P. Agarwal ma'am, O.P. Agarwal sir, Managing Director, Dr. Nima Agarwal ma'am, Additional Managing Director, Sri Raman Batra sir, Executive Vice President of NIT, for their continuous support, which have greatly helped towards successful organization of this conference. It is my immense pleasure to express sincere gratitude to our chief guest, Dr. Sailendra Saraf sir, and the guest of honor, Dr. Atul Nasa sir, for kindly accepting our invitation in spite of their busy schedule and gracing the inaugural function. I am sure that all the participants have taken note of your interesting and knowledgeous address. Really, sir, we all are inspired by your great words, and mainly the whole effect of antibiotic on the ecosystem along with the human beings. We also acknowledge our gratitude to Director General NIT, Professor Praveen Suneja sir, and Director NIT, Dr. Vinod M. Kapse sir, for their inspiring mentorship and gracing the inaugural ceremony of this e-conference. Well, an event like this cannot happen overnight. It requires proper planning and a bird's eye on detail. We are really fortunate enough to be backed by very motivative and dedicated Director of NIT Pharmacy Institute, Dr. Abhijit Majumdar. The initiation and enormous hard work of whom made it possible to be a successful e-conference. We are also really opportunistic to get the continuous research guidance from our Dean Ma'am, Dr. Rupa Majumdar Ma'am. We owe our thanks to our Dean Ma'am. And I would like to express our heartiest thanks to all delegates for their enthusiastic participation in the inaugural ceremony of the conference. We have noted your queries and and we also appreciate your involvement in the e-conference. I acknowledge the reviewers, the technical committee, the organizing committee, and the registration committee members for providing their valuable support and suggestions on time. I also acknowledge all faculty and staff members of NIT Pharmacy Institute for their support to organize this inaugural ceremony and the whole e-conference. We are thankful to all HODs, deans of various departments, Registrar NIT for their valuable suggestions and support to make this conference possible. We acknowledge our thanks to media, Press, Accounts Department NIT for their constant support to organize this e-conference. I acknowledge the unwavering support of Mr. Vikas Tiwari EN, Department NIT and other technical staff for their valuable technical support for the smooth conduction of the e-conference. I once again extend our thanks to everyone whose contribution made this conference as a successful e-conference. Be safe, be healthy. Thank you, sir.
Thank you, ma'am. Thank you for your nice vote of thanks. Now, I would like to request you all for the national anthem. And just after the national anthem, we'll start our technical session 1. So be with us.
गणपनमधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंग विंध्य हिमाचल यमुना गंगा उज्ज्वल जलधि तरंग तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
So, Nasa sir, we are meeting 3 o'clock. Okay, sir. Balla sir, Dr. Mazumdar.
Right,
okay.
Thank you very much.
Thank you, sir.
Thank you.
Thank you, sir, for joining me. Thank you. Stay safe, sir. Take care. Now, I would like to start our technical session 1 by welcoming Dr. Vijay Bhalla, sir, for addressing us regarding the theme of the today's conference. Sir has already joined us. Professor Dr. Vijay Bhalla is working as Principal SGT University, Gurgaon. He is having 34 years of experience. His area of interest is pharmacovigilance and social administrative pharmacy. He has more than 100 national and international publications of repute. He is also working as president of IPGA and he was the chairman of 60th, 65th and 70th IPC. He is recipient of Excellence Award, Best Dean SGT University, then IPGA Professional Excellence Award, IPGA Fellowship Award. Sir, I would like to request and welcome you, sir, to address our participants and delegates. Bala sir, please.
Am I audible, Dr. Somya?
Yes, sir. You are audible. Please switch on your camera, sir.
Yeah, it is on now. It is on now.
Yes, sir.
Are you able
Yes,
to see my slides?
not
Are you
yet,
able
sir.
to see PPT?
No, sir. Not yet.
No, not yet. I'll check up. Just a minute.
Sir, try once more sharing.
Yeah,
Once again, you can try.
Is it okay?
Yes, sir.
Now,
It's
visible?
fine, sir. Yes, sir.
Now you can see?
Yes, sir.
Okay, great. So good morning all. In fact, first of all, thank you very much to the organizer, particularly Noida Institute of Engineering and Technology. I have a very long association with NIT since 2006. And I'm very much close to our Professor Abhijit Majumdar, Dr. Rupa Majumdar, and Soumya Das, Sanjita Das. You are doing a good job. Thank you very much to the management also for giving me an opportunity to talk on such a burning issue on antibiotics and resistance, a pharmacist viewpoint. I am also thankful to Dr. Shirendra Shroff and Mr. Atul Nasa. We heard them as a chief guest and guest of honor. More than that, Dr. Shirendra Shroff is an advisor to central government by the route of Drug Technical Advisory Board. And Mr. Atul Nasa also is an advisor to state and central government by the route of Drug Consultative Committee. These are the two major committees. So I am seeing Noida Institute of Engineering and Technology growing from such an inception now to an NIRF ranked and NAC accredited institute. It is one of the very fine institute in Greater Noida region with the leadership of Dr. Majumdar and a very good team of the faculty. A lot many dedicated and learned faculty members are there. So I am very happy to, in fact, be a part of this seminar and this conference, 3 days AICT sponsored conference. So topic is very interesting. We have heard a lot from many people, and I think by core of our heart, we all know also many things about antibiotics also, about resistance also, and the contribution and role of pharmacist also. But I would like to once again sensitize you all people where we are actually heading. So this topic is of great interest to me also as a person, because I have spoken earlier also in these topics, and I am trying my level best wherever possible in the community to sensitize the people. From IPGA front, we have released some of the leaflets also on this issue for the community pharmacist. So let's hear on this. I think most of know, and Corona has also made us to understand to say namaste over handshake. Because by saying namaste, we do not transfer germs while greeting our friends. We have seen, one study has revealed that by shaking our hands, we transfer 124 million bacterial colonies to one another. If we say high five, we just High five is basically we are clapping hand with one another. We are transferring 55 million bacterial colonies. If we are just first bump, 7 million bacterial colonies are transferred. And by Namaste, we are doing a zero bacterial transfer. So I think our culture of Hindus, our Indian culture is too good. And I think now the world has also adopted this Namaste. So we have setting the standards and we are remaining hygienic. And I am very sure this is going to reduce the bio load also. So now Corona has also further intensified this campaign by washing our hands repeatedly with soap and water. So I think that has also helped us to maintain hygiene. And if we are able to maintain hygiene, I am very sure that the bacterial burden will be reducing in time to come. I think a very interesting study I was going through in the COVID time, the first time in Indian history, the pharma market scaled very high sales of diabetic and hypertensive drug, and the sales of antibiotics was reduced. It was on a second and third number. Otherwise, before February, every month, if you check and track the sale of antibiotic was higher. So probably one of the reason, if we are maintaining hygiene, some of the common infections we are not getting trapped in. Now, online webinar challenges. We are in a technology era. We have to adopt technology very fast. So some of the challenges, eye-to-eye contact, expression and emotions are missing. Otherwise, I would have enjoyed this session. Technology issues, sometimes bandwidth problem could be there. I am not audible, you are not audible, you can't see, I can not see. So these are there. Motivation and time management is missing sometime. Adaptability struggle and computer literacy to some of the seasoned and elderly people can be noticed. And many a time the attended feel isolated and disconnected, and they get a non-serious feel also. So I think these are the online webinars challenges. But even after that, I think we are growing very fast, and these type of challenges we have to accept. And I think we should learn these technologies and we should be sharp enough. So the very first slide of mine, a very powerful dialogue by Shah Rukh Khan: don't underestimate the power of a common man. So I think now it is a battle of a common man versus a microbes. A common man can really protect himself from the microbes, but microbes once invaded in a person, I think they will make colonies and colonies, and they will not try let free that space. So I think we have to boldly engrave this statement that don't underestimate the power of microbes. If they have invaded you and if they are pathogenic in nature and their virulence is noticed, I think then we are the victims. Very interesting study recently I was going through. The COVID-19 could reverse decade of progress towards eliminating preventable child deaths. Agencies warn, like WHO has warned, that we are behind 10 years just because of this COVID-19. So our growths are hampered. Another interesting study, I think Dr. Shailendra Shroff has also made a mention of it, that nearly 2 million Americans per year develop hospital-acquired infections, resulting in 99,000 deaths, and majority of which are due to antibacterial resistant pathogens only. And out of these pathogens, 2 common hospital-acquired infections alone are sepsis and pneumonia. I think pneumonia, COVID also is getting into one type of pneumonia only. So these are the 2 major infections basically where we can definitely see that many deaths are there. And this is America, who is maintaining a digital records. I think in our country, we do not have a record. So the number of deaths would be much higher in country like India. So what are antibiotics? I think just to recapitulate, these are the powerful medications or medicines with one very important job to fight against disease causing bacteria. When used properly, antibiotics can save lives. But bacteria that mutates and are able to resist the antibiotics, that are meant to kill them. So this is a power of microbes. They can mutate. This is a normal process speeded up by the overuse and misuse of antibiotics. Mutation is a normal process, but it can be speeded up by some of the activities we undertake. Some subpopulation of microorganisms are able to survive even after exposure to one or more antibiotic. These were those mutant strain. So microbes develop resistance to the antibiotics. Pathogen resistant to multiple antibiotics are considered multi-drug resistance. Like we know, MDR tuberculosis is very common. People are resistant to rifampicin, pyrazinamide, isoniazid, ethambutol, streptomycin. So many of the 2 drug, 3 drug, 4 drugs, they are resistant. So that condition is called a multi-drug resistant basically. Then we also hear these days a term, even in tuberculosis only, XDR tuberculosis, excessive drug resistance, which is very rare condition, and I think these type of patients are very difficult to treat. A very interesting remarks by finding by WHO that resistant threat, basically treatment of a ever increasing range of infection caused by bacteria, parasite, fungi and viruses. So threatens, AMR threatens, resistance threatens basically. These are the difference: antibiotics and antimicrobials. In antimicrobials category, you will get all type of microbes, basically parasites, viruses and fungi. AMR occurs when bacteria, virus, fungi and parasites change over time and no longer respond to medicine. Antimicrobial, including antibiotics, antiviral, antifungal, antiparasitics, are medicine used to prevent and treat infections in human, animals and plants. So microorganisms that develop antimicrobial resistance are sometimes referred to as superbugs also. We commonly hear these terms, superbugs. Even computers these days have a viruses. So human viruses are very dangerous. They are called superbugs. How do we get this resistance basically? If a person gets a very minor infection, which is a primary infection for the first time, and he has been prescribed one antibiotic by one of the learned physician, but that patient, if he doesn't consume, if a inadequate antibiotic treatment or poor compliances are there, then there are the chances of resistant infection. Bacteria cause of the infection are eliminated and resistance bacteria are developing. So once that condition comes, then we give another antibiotic, antibiotic number 2, 3 or n number. So the proliferation of bacteria causing disease that are more serious and more difficult to treat. So now we are left with a very highly sensitive and sophisticated antibiotics. So they can heal you or they may not heal you. If they don't heal you, then we are at the therapeutic deadlock. In some cases, bacteria become resistant to all treatment known to date, so that patient finally has to get into a septicemia type of conditions, and multiple organ failures are reported. Some of the key facts given by WHO: antibiotic resistance is one of the biggest threat to the global health, food security and development. Antibiotic resistance can affect anyone, any age and any country. Antibiotic resistance occurs naturally, but misuse of antibiotic in human and animal is accelerating the process. A growing number of infections such as pneumonia, tuberculosis, gonorrhea and salmonellosis are becoming harder to treat as the antibiotic used to treat them becomes less effective. Antibiotic resistance leads to longer hospital stay, so higher burden, financial burden, higher medical cost and increased mortality also, beside morbidity for family. So we are thankful to Alexander Fleming. I think he was the first person who gave an antimicrobial, penicillin, in way back in 1923. But today, if Sir Alexander Fleming would have been observing that we people have really destroyed his dream, because just after 1923, when he discovered, in 1928, the penicillin was marketed and there. So 1940 only we received the dates of resistance identified for penicillin. 1948, whatsoever the antibiotics were like tetracycline came in the picture. Even some of the macrolides came, erythromycin type of category of drugs. So in 1953, we found a resistance to these antibiotics also. So then in 1985, we saw that carbapenem, one of the very latest generation of antibiotics came. So even fluorophenols came in that category only. So even these days also we are finding resistant to these highly sensitive antibiotics. So truth is antibiotic can be bought without a prescription in many countries, and overuse of the drugs is fueling the evolution of resistance microbe. Mr. Atul Nasa has recently told. We are, as a pharmacist, I think this crime we are doing. So we have to stop that crime, because we should not sell any of the antibiotic without prescription. Some of the AMR worldwide, 14,000 patients die in USA with Clostridium difficile only. 23,000 patients die each year in USA due to microbial resistance. 2 lakh infections with contains bacteria that are resistant. 11,000 deaths each year due to
Sir, you are not audible, sir. Excuse me, sir. Sir, please unmute yourself. Sir? Sir, you are not audible, sir.
Am I audible?
Yes, sir.
Yeah.
Now audible.
Okay, okay. PPT is there?
Yes, sir.
Okay. So somebody unmuted from that side only.
Okay. Okay, sir. Sorry.
So Indian statistics, if we look at, according to Public Health Foundation of India, India records 70,000 new multi-drug resistant tuberculosis cases. Even second line artemisinin for malaria. 5% of all HIV/AIDS cases are drug resistant, and there are also the larger burden of drug resistant bacterial infection, especially in hospital. Now let's look at the causes, major causes. I think some of the causes have already been highlighted, but these 4 categories has been downloaded. The pre-existing resistance population of bacteria in the nature. Now even nature has n number of resistant bacteria. So if we are invaded by them, then it is our ill luck. Inappropriate antibiotic use. Microbial exposure to all antibiotics. We are trying hit and trials with antibiotics for our treatments. Microbes have most likely invented antibiotics against every biochemical target that can be attacked. Even microbes are playing smarter. So what is the urgency? Antibiotic resistance has been declared a crisis by WHO, Center for Disease Control and Prevention, Institute of Medicine, Infectious Disease Society of America, National Communicable Disease Center in Delhi, virtually all other relevant organizations, that they are posing terrorism on the list of threat by resistance to antibiotics. So this is one of a great mandate. Professor Dame Sally Device says if you get an infection in your bloodstream, in about 10 to 20 years, it might be untreatable. So now let's go to interventions. What type of interventions we should look as a pharmacist? So these are very important. I think one must concentrate on these interventions. If we intervene by this type of interventions, I'm very sure in time to come we will have a control over resistance. The very first intervention could be preventing infections and resistance. That is health, hand hygiene, we are playing very safe. But self-cleaning, disinfectant by mist or vapors or radiation, they are still clinical validations are not there. So clinical validation is required in these cases. Preventive infections, and like we see even in advertisements, Dettol's and many other disinfectant claim 99.9, but clinical validation is yet not there. I think our government policies has to reframe. Novel delivery systems are to be adopted, like drug delivery or IV catheterization, prosthetic or non-invasive ventilator. So these type of therapy should come where we are not directly contacting the human being, so that we can prevent contamination and infections, but still under conceptual stages. Improvement of population health to reduce readmission to the hospital. It is an implementation stage. We should not, at least the readmission, that is morbidity, mortality, if it is increases, because from hospital we are many a time acquiring the infections and we are getting readmitted in the hospitals. Some of the niche vaccines are to be developed to prevent resistance bacteria. So we have to work on now to look into the specific bacteria. Another intervention which is very important is refilling antibiotics pipeline by aligning economic and regulatory approaches. Dr. Shailendra Shroff told that our pipeline is dry because companies are not spending money on the investments on the short course type of therapies. Antibiotics are given for a shorter course, 7, 10, 15 days only. So they compare commercial viability when it is compared to dyslipidemic agents or anti-diabetics or anti-hypertensive, where they are commercially very stable. So that is the reason our antibiotic pipeline is really dried up. So there must be funding in research and development to develop antibiotics. So models are in place, funding is required. These interventions much required. Institutions of novel approval pathway for limited population antibiotic drug proposals. I think our regulatory actions are required in these cases. Some of the specific institutions must given on the approval for limited population of antibiotic drug proposals. So our regulatory has to be sympathetic to give clinical trial recommendations and proposals approval. The next category of interventions is preserving available antibiotics. If we are not able to preserve the available antibiotics, I have seen and observed many of the physicians, old-time physicians, still use sulfamethoxazole and trimethoprim, that is Saptran combination. They don't go for cephalosporin or macrolide type of antibiotics. They are still, and those antibiotics are still working. So, but our physicians also has to be sensitized not to prescribe a higher category of antibiotics, and our pharmacists also should not dispense it on their own. So public reporting to antibiotic use has to be increased. So policy action required. Development of rapid diagnostic and biomarker test. I think we are missing, because ours is a country, we never go for a culture sensitivity test for the antibiotic usage. We don't know which antibiotic is really useful and working very hard and fast and quick. So I think some of the diagnostic and biomarker tests are to be advanced, so that within 24 hours, 72 hours, we are able to identify the strain of the microorganism, and we are able to do some culture sensitivity studies, and we are able to give a perfect weapon, I think, to kill that microorganism. Elimination of use of antibiotics to promote livestock growth. I think we are still in our country using hell of a lot antibiotics for poultry farming and other animal husbandries. I think there has to be elimination. Some legislation is required where the use of antibiotics for poultry farming and livestock growth should be really curtailed. New waste treatment strategies. I think Mr. Atul Nasar touched upon the waste treatment. We are really throwing away some of the unused medicine, expired medicine. So those new waste treatment strategies are to be really framed. Some policy implementation should be there. WHO has given a guidelines. National Formulary of India developed by Indian Pharmacopoeia Commission is also given some guidelines. I think we must adopt that policy and implement as such immediately. Studies to define shortest effective course of antibiotics for infection. So I think trials are required in these cases. So we should have a shortest effective courses. Maybe some MIC levels are to be again retried or repurposed. So some of the antibiotic are to be repurposed, that if we can curtail the longer duration of 7 to 10 days to a 2 or 3 days, I think we can still control some. Another interesting intervention we required is a developing microbe attaching treatment with diminished potential to drive resistance, like immune based therapies, like we are developing vaccines for corona also. So research is required, funding is required. Antibiotics that do not kill bacteria but alter their ability to trigger inflammation or cause disease, I think there again it is research required. I think some Cadila is working on this. We have to really make some intervention in the area of immunotherapies. Developing treatment attacking host target rather than microbial target to avoid selective pressures, driving resistance. So you have seen the cases of corona. I think we are in a state of hyperinflammatory state, okay, which is also called cytokine storm or cytokinin surge. Because of that hyperinflammatory state, I think multiple organ failures are taking place. So we have to develop some treatment attacking host targets rather than microbial targets. So we have to work on that area also, how we can reduce the cytokine storms or inflammatory exudates. Direct moderation of host inflammation in response to infection. So cytokinin agonist or antagonist or pathogen associated molecular pattern receptor agonist should be developed. So proof of principle stage is already on. Some of the companies are and some of the research organizations are working on it. Sequestration of host nutrient to prevent microbial access to nutrient. So we can also try this research also, proof of principle concept, how we can really prevent host nutrient so that the microbial access to nutrient is diminished and microbes are not able to grow. Probiotics that compete with microbial growth, we have to develop some of the probiotic if they are able to competitive antagonistic approach type of thing. So probiotic approach can be developed. So some of the suggestions looking to this is economic and regulatory disincentives have made it far too difficult for companies to continue developing new antibiotics. There are no regulatory incentives are there. So the government need to incentivize pharmaceutical companies and other research institutions also to develop new antibiotic, because it is not happening right now. Agriculture and farming industry need to take responsibility for the antibiotics that they use every day. So they should not use it, agriculture and farming industries, because the traces lying in their areas are developing lot many resistant microbes in the nature. So with which I think we all people are getting invaded and infected. So threat could be 10 different antibiotics can be used to treat a serious E. coli infection. But if we do not make it our problem now, it will certainly be our problem later when we are asked that antibiotic to use for a pan-resistant E. coli infection, and the answer would be nothing. In spite of having 10 different antibiotic today, if later we come across that no antibiotic is working in E. coli, I think that is the greatest threat we have. Antibiotic resistance is putting the achievement of modern medicine at risk. Organ transplantation, chemotherapy, and major minor surgeries such as cesarean sections becoming more and more dangerous without effective antibiotics for the prevention and treatment of infection. So if we do not have a strong antibiotic in the pipeline, even these surgeries would be very dangerous in future to come. So let's just summarize the things. Some of the causes at the pharmacist and community level over and use and abuse of antimicrobials or antibiotics because of poor prescription, lack of adherence to the prescribed medicine. Patient is demanding from the chemist, and economic incentives are given by the companies to selling more product. So we have to develop new antimicrobial, no longer depending on these options, because very few new agents are there in pipeline. So we have to definitely reserve these antibiotic for the future generation and future infection. Unregulated availability of antimicrobials are there. I told some of the enforcement. I would like to really stress upon this, Atul Nasa sir, enforcement has to be stringent so that it is not available freely. Poor quality medicines are one of the reason. Inadequate infection control in the hospital is another reason. Inadequate healthcare system to manage antimicrobials. Lack of knowledge by prescriber and user, and pharmaceutical companies promotion policies. These all are really causes of developing antibiotic resistance at the community level. So preserving effectiveness of existing antimicrobial by rational using, by infection control strategies, and implementation of Schedule H1 regulation. Mr. Atul told you that H1 regulation has been devised. What is H1 regulation? In H1, some of the approximately 47 drugs has been transferred from Schedule H to H1. And in H1, there are 3 categories of drug, all anti-tuberculosis drug, major habit forming drug like alprazolam, clonazepam, and all 3rd generation cephalosporins are included in the H1 schedule. So these 3rd generation cephalosporin, we see a whole range of cephalosporins, but nowadays they are also not very effective because the resistant strains are available. So key prevention strategy in total comes out to be preventive infection and transmission. We can prevent infection and transmission. I think mask is one of a great asset these days people are having. I think many of the infections can be prevented from transmission if we are using mask on our face. Effective diagnosis and treatment, like some of the culture sensitivity techniques, optimizing use of antibiotics like the prescribed days. Immunization programs are very important, and insisting on hand hygiene. I think that should be a regular practice by each and everyone. So let's visit our global action plan of WHO. The global action plan on antimicrobial resistance has 5 strategic objectives by WHO. Number 1, to improve awareness and understanding of antimicrobial resistance. I think every person should be aware of this, and community pharmacist has a great role to play because they are the neighborhood chemist, and they have to really make the public aware not to use antibiotics rampantly. To strengthen surveillance and research, surveillance by the enforcement people, regulators, and research for bringing newer antibiotics, to reducing the incidences of infection in hospitals particularly, to optimizing the use of antimicrobial medicines. Like each hospital should have a therapeutic committee, and that therapeutic committee should not permit the use of 2nd or 3rd line of antibiotics. They should only allow the physicians to use the 1st line antibiotics only in initial primary infection conditions. So to ensure sustainable investment in countering antimicrobial resistance by virtue of giving incentives to the pharmaceutical company for developing these type of products. So I think I have tried to cover my over to Soumya. I think
Yes, sir.
some of the basics.
Very nice, sir. Very nice, sir. You described and discussed in very detail, sir.
Thank you so much. Thank you. I hope people have enjoyed it and understood. Still, they have any type of questions, or you note it down, or you can ask me, or you can get the information from me anytime. I shall be available for you people anytime.
So there is one question from the audience side.
Yeah.
They ask, sir, why the word exact the term antimicrobial is used. Uh, the word antimicrobial, but microbes are wide variety of organisms, and why
Yeah.
we are talking only about antibiotics.
Look,
The question is,
there, yeah, actually the problem is what humans are using. Antimicrobes are of plenty of types. Like people are using antimicrobial agents in agriculture industry. People are using antimicrobial agents in many other form also. But in context of human usage, we are dependent on most of the antibiotics. The predominantly antibiotics are being used by human beings on prescription. A very less chemical synthetic antimicrobial agents are available. Most of the drugs which we obtain from the biological sources only, they are being used. So these antibiotics are also fall under the category of antimicrobials only. But antibiotics basically are, since they are the origin, their origin is from biological sources, and we are very much dependent on. So their resistance is of great concern to a human being because they are taking lot of deaths and which are accounted for.
yes, sir. Thank you, sir. Sir, one more question. How can we help as a layman to tackle the antibiotic drug resistance?
As a layman, the very first is, I think as a layman, we should, number one, we maintain hygiene to the utmost of our efforts. Simple human hygiene. I think from where you try to find out the entry point of any of the contamination or infection. I think COVID has sensitized each and every person of our country that the entry point of any infection is either from eyes, nose, or mouth, and hands are the culprits. We take our finger, we rub with our eyes, we rub our nose, we take those fingers to our mouth. So if we start maintaining hand hygienes by running water and soap, I think 50 to 70% of the contamination we can really avoid. And these contamination are the only reasons. They start with primary infection. Once the infection loads into, we are given or we are prescribed antibiotics. Once the antibiotics are prescribed, then we underuse, misuse, overuse, our compliances are not there, then we are trapped into a resistant type of category. So I think we have to safeguard ourselves from the primary contamination. So if we are able to save ourselves from primary infection, I think the purpose would be solved to a greater extent. Hello.
Yes, sir. Sorry. Sir, one more last question.
Sure.
What is the single most significant factor driving increasing rates of antibiotic resistance?
I am telling you, if we see the doses adherence is one of the, as a pharmacist viewpoint, if I'll say the compliance and adherence dosage is one of the single factor which is really driving us to a greater, because once we are infected and invaded by any of the organism, we have to have some medicine. We cannot get escape. But the problem lies there. We are not following religiously the treatment protocol advised by our physicians. So probably one of the single, from the pharmacist point of view, is that reason only. And I think TB is one of a burning example that MDR and XDR tuberculosis is growing faster because people are not really adhering to the schedule. I have seen people getting cured from ATT regimen who have religiously followed the treatment. So I think as a pharmacist, we must encourage each patient to use antibiotics religiously for the time till the symptoms disappears.
Very true, sir. Right, sir? Thank you, sir, for your very nice and detailed, informative talk, sir.
Thank you very much.
You very clearly in detail described about the global prevalence, the various major causes of these infections, about the super bugs, how we can get these infections, then how one should develop the mechanism of drug resistance, then poor patient compliance, WHO global plan, and then the role of community pharmacist, how they can show their responsibilities towards this global problem to overcome this, and how one can strengthen the surveillance as well as research towards this global problem. Sir, I request you to create a social awareness by percolating this message in the society through members of IPGA, sir, through IPGA Students Forum. Thank you,
Sure,
sir.
sure. Thank you so much. Thank you so much. If you have
Stay
a
safe,
time,
sir.
I have a small video on World Pharmacist Day. That just 2 minutes video. If you have a time, can
Sure,
I play?
Sure, sir. Sure.
Okay. Just a minute. आया वीडियो आ गया क्या सौम्या?
नहीं सर, नहीं आया सर अभी.
नहीं आया. Just a minute.
Okay, sir.
Just
no problem, sir. Your time.
आया भी नहीं.
नहीं आया सर. It's not shared, sir.
Not shared. Just a minute. एक minute. Actually, videos sometimes take time to.
Yes, sir. We have time, sir.
No problem. No.
किसी वो कर रहे हैं हम लोग.
Just a minute. हाँ. सूरज एक मिनट आइयो, ये वीडियो शेयर नहीं हो रहा जरा. Just a minute. Just 2 minutes. आ गया क्या?
नहीं आया
सौम्या
सर.
आया क्या वीडियो? नहीं आया.
नहीं आया सर. नहीं देख रहा हूँ.
Just
Not.
इसमें शेयर करियो. अब आ गया?
Yes, sir.
आ गया?
Yes, sir.
इसको शुरू से करें. Just a minute. आ गया?
Yes, sir.
Okay, good. ये ना वॉइस भी आ रही है ना थोड़ी?
वॉइस नहीं आ रही सर.
क्या करूं?
प्लीज शेयर ऑडियो सर.
क्लोज कर दिया था ना. हैं?
आ रही है वॉइस?
नहीं सर, नहीं आ रही है। इट्स नॉट कमिंग।
तेज करिए। हैं? क्या चीज? मेरे को म्यूट कर देना। मेरे को हाँ, मेरे को आ रही है। आपको तो आ रही है, ये बोले चला रखा है ना। नहीं, इसमें से आ रही है मेरे को। तो वो आपको तो आएगी, उनको आ रही है कि नहीं, या इसमें देखिए ये कर आ रही है।
It's not audible, sir.
It's not audible. Just a minute.
चल बंद कर दे। चलो कोई बात नहीं, छोड़ो। ये अब आया क्या? चलो, आया था सौम्या? नहीं। हेलो।
Sir, you can send us. We can share with the participants.
हाँ ठीक है, ठीक है। I'll mail you. मैं व्हाट्सएप कर देता हूँ अभी मैं थोड़ी देर में। ठीक है।
Okay, sir. Okay.
Thank you very
Thank you,
much.
sir. Thank you, sir. Take care, sir.
So okay,
So,
I'm leaving the meeting now. ठीक है।
oh, sure, sir.
कहाँ से?
I'm requesting to all the participants and delegates to be or join with us at sharp 2 p.m. after the lunch break for the oral session. Thank you.
सर प्रेजेंट कौन कौन है?
Good afternoon, dear participants, respected judges, to the oral presentation, which is the second half session of AICT sponsored e-conference on antibiotic resistance, causes and future challenges in digital trial. I welcome Dr. Rupa Majumdar ma'am, Dean, NIT Pharmacy Institute, Dr. Rasmi Mishra ma'am, HOD Biotechnology NIT, Dr. Salahuddin, HOD Pharmaceutical Chemistry, NIT Pharmacy Institute, as a judge to judge the oral presentations of the participants. Thank you so much. So I would like to invite Asiru Anis Yayu to present his oral presentation. Asiru Anis Yayu, please. So judges, please judge this presentation first and we'll go. Ashiru, are you there? I think you have joined, but is there any problem to show your slide, show your presentation? Ashiru Anis, if there is any problem, you can inform us so that you can present afterwards. So Ashiru, I would like to inform you that you can present afterwards. We should proceed, and Manish Kaushik, please present your presentation. Manish Kaushik. Now you can share your slides and you can present. You can start your presentation. Manish Kaushik. Yes, Manish.
Hello.
Yes. Yes, you are audible, quite audible.
Okay. Ma'am, I am Dr. Manish Kaushik, Institute of Technology. And thank you
Manish, your,
to Farm. Hello.
yes, Manish, you can on your camera.
Just a minute, ma'am.
Yes, yes,
It's a
good,
bit.
good. Very good. Please carry on.
Just a minute, ma'am. It's a bit slower over the internet. Okay. So
Not a problem.
my
Take it.
today's topic for the presentation is functionalized nanomaterials based biosensor for detection and diagnosis. First, I would like to give a brief introduction why these biosensors are used. So biosensors are very significant for the identification of target molecules with high accuracy, selectivity, and a good signal to noise ratio. So it provides a better way to identify the target molecule. These biosensors are used to detect biomolecules. For the same, enzyme and nucleic acids are used as probe. Nanomaterials like carbon nanotube, nanocomposite, quantum dot, and gold nanoparticles are used in preparation of these biosensors. These biosensors also helpful in screening of metal ions, etc. So first we would like to know what is biosensor. Biosensor is a device which responds to chemical species in organic sample prepared using the biological component, or any device which is prepared using biological component are called biosensor. It may be used to sense some inorganic compound as well. A biosensor seems something like this. A analyte would be the sample which provide a biological signal. In biosensor, there is a bioreceptor part. Bioreceptor part detect that signal. Transductor may be a nanomaterial which translate that signal to the receiver, and then the signal is converted into a display or any signal processing unit. It displays the reading value. So a sensor integrates the biological element, physicochemical transductor, and produce a electronic signal. So these are three parts: analyte, bioreceptor, maybe enzyme, antibody, microorganism, etc. Transductor may be detect any electroactive substance, pH change, heat, light, or mass change. This transductor create, by these change it provides some signal, and these signal are detected by the detector unit. So first component is the biological element. So biological element is the component which is used to bind with the target molecule. It must be highly specific, stable, and under storage condition it would be immobilized. Microorganism, tissue, cell organelle, nucleic acid, enzyme component, receptor, and antibody can be used as this part. Second part is physicochemical transductor. This acts as an interface measuring the physical change that occurs with the reaction at the bioreceptor and transform that energy into measurable output. So this output may be an electrical output, any pH change, heat, light, or mass change. I will discuss them one by one later. Third component is the detector, which detects the signal from the transducer and passed into microprocessor where they are amplified and analyzed. And this data is then converted into concentration unit and transferred for the display. There may be 4 types of detection signal which are produced by transductor. It may be electrochemical signal, which gives the information of any change in electrical charge. It is due to the movement of electron in redox reaction. The transductor may give optical signal. This is due to the change in light intensity because of absorption or emission of light. It may give thermal signal. It would be due to the change in temperature. This change in temperature because of the exothermic or endothermic enzyme catalyzed reaction, or piezoelectric effect, which is showing by the change in the mass. So Piezoelectric materials are like crystal which vibrate under influence of electric field, and due to these vibration, a slight change in mass is observed. So piezoelectric signal can also be produced by a biosensor. My paper is a mini review article in which I have reviewed few recent advances in biosensors. So few biosensors based on carbon nanotubes are prepared for the detection of hydrocarbon in water. For a multi-wall carbon nanotube based biosensors are prepared to detect lead in water. For detection of glucose, lactic acid in body are also prepared, and for the detection of tumor, the biosensor are also prepared. So in a biosensor, there would be a antigen, this is carbon nanotube, and a antibody which is acting as the biological part, a biological sensor which sends the information, and then transductor transfer the data to the receptor part. Gold nanoparticle based biosensors were also prepared for the detection of protein and nucleic acid, and these biosensors are extremely sensitive up to the concentration of 20 attomolar concentration. 1 attomolar is about 10 to the power minus 18 molar concentration. Gold nanoparticles for the detection of viruses are also prepared. Gold nanoparticle based biosensor for detection of pesticides are also prepared. Quantum dots based biosensors are also prepared for the detection of microorganisms, for detection of cancer, for detection of tumor necrosis cells at 60 attomolar concentration, for detection of other proteins of microorganisms. These biosensors are very helpful. So it may be possible that few person are already aware about the biosensors, but I have few daily life example of biosensors like glucometer, by which we check our glucose level by touching a drop of blood with it. The pregnancy test kit, ovulation kit are also the example of biosensors. These are based upon nanomaterials. These are the summary of few highly reactive, highly sensitive biosensors which are prepared and referenced in my paper. Are the references. Thank you very much.
Thank you. Mr. Manish Kaushik, your presentation was on functionalized nanomaterial based biosensor for detection and diagnosis. Okay. So yes. My question is, how will biosensor work in case of tumor targeting or identification? You can cite one live example also if possible.
Yeah. Yes, ma'am. Ma'am, in tumor cells, there are secretion of few enzymes, and those enzymes are detected by these biological sensors, and it works at higher sensitivity up to 60 attomolar concentration. Even at 60 attomolar concentration, it gives a positive signal. So we can detect cancer at very primitive stage.
Only enzymes will be detected, or expressed proteins also can be detected? Yeah. Achha, my second question is that how will you combine these three parts which you have told, bioreceptor, transducer and signal processing unit?
Ma'am, these are first when we try to prepare a biosensor, we select these part which have a reactive site over there. So as we can see that gold nanoparticle, carbon nanotube, or quantum dots already are a few compounds which already have bonding sites or functional site. So by virtue of these functional sites, we easily combine them together.
Thank you from my side. And I think other judges will also ask you some questions.
Yeah, sure, ma'am. Thank you, sir.
Hello. Good afternoon, Manish. This is Rashmi
Good afternoon.
from Biotech
Good afternoon,
Department.
ma'am. Good afternoon.
Manish, I had a question. Like you have the nanoparticles that was generated
In the development of these nanoparticles, we are working towards the preparation also. Right now, I have written this a review article for this seminar.
So what is the effective size of nanoparticle that is coming in your studies?
Ma'am, nanoparticles are considered for the term nanoparticle is considered for the products which are at 1 to 100 nanometer scale, and
True, that is true. But what was the effective size that you are studying?
Sorry, ma'am.
What is the effect? What is no issues? I just wanted to know the effective size that you are going for biosensors.
Okay, ma'am. These biosensors are used on basis of carbon nanotubes, which are a size of 100 to 200 nanometer size. These are of 100 to 200 nanometer in diameter, and the gold nanoparticles were of 60 to 70 nanometer size, and quantum dots also
Manish.
of 25 to 95 nanometer.
Thank you, Manish. Thank you so much.
Hello.
Hello, sir.
Manishi, I have one question. Which type of detectors used in, you have given the example of glucometer as well as the pregnancy test kit. So which type of detector used in this instrument?
So these detectors are based upon some electrical signal. So electrical detector are also being there. Sometimes it gives the color change. So the detector which gives the change in intensity of color are also being there. So it may be on basis of the developer which type of detector he wants to use.
There is no chemical reaction occurs in between this
Yes, sir, definitely. The transductor are based upon the chemical reactions. Those chemical reactions occur inside the processing. Because of these chemical reaction, also the signal is produced.
Okay, Manishji. Thank you.
Thank you. Thank you.
Manish, I want to know the name of your institution.
Ma'am, I am from Noida Institute of Engineering and Technology.
Acha, NIET.
Yes, ma'am.
Okay, thank you.
Thank you, Dr. Manish. Now I would like to our second oral presenter, Mrs. Bhavani Pentela from NIT.
Good afternoon, everyone. Am I able to hear?
Yes, you are audible.
Thank you. Thank you. So, good afternoon to each and everyone, my colleagues, participants, and respected judges, and my dear students who are all here. And first of all, I have to thank to Director Sir, Avijit Majumdar Sir, to giving me such an opportunity to present here. And let me share the screen. मेरा screen is visible.
Yes, it is visible. You can carry on.
Thank you. So today my presentation is on current progress, impacts and challenges of drug-resistant superbugs on COVID-19 pandemic. So before going to the topic, let me see the definitions which involved in this review articles. So first one is antibiotic. So everyone knows what is an antibiotic. So it is a drug which is used to treat the microorganisms. Either it will kill the microorganisms or it will inhibit the growth of microorganisms. So what is necessity by giving the superbugs here? Why I had related superbugs to the antibiotics? Because जी sir.
Yes ma'am, इसको full screen कर दीजिए.
Sir, 1 second. Sir, Abhi, now.
Move on to full screen, ma'am.
Sir, full screen कर दिया sir. Slide.
It's okay. Now it's.
Sir, now is it okay?
Yes, it's okay, ma'am.
So, what is meant by superbugs? The superbugs are the strains of bacteria, virus, parasites, and fungus, which will produce the resistance to the bacteria, which will produce the resistance to the antibiotics, for which we are going to treat here. And mostly, we can say antimicrobial resistance, sometimes referred as superbugs also. And these superbugs will cause us resistance to the antibiotics whenever we have given for the infection. And next, introduction to these antibiotics and superbugs. In recent years, antibiotics has been losing their punch against some types of bacteria. In fact, certain bacteria are now unbeatable with the today medicine. Because if you see here, a lot of articles, they are representing and presenting that the 7 lakh lives due to the drug resistance superbug, we are losing almost 7 lakh lives for every year. If it may continues, if the drug resistance continues, by 2050, the death will be around 10 millions. Among 10 millions, the 2 million people will be our Indians, because the Indians are one of the biggest consumers and producers of the antibiotics. So it may the growth matlab yahan the death rate will be increases while we are taking the antibiotics in a more quantity or misused quantity. And how we'll get the superbug resistance? Because of an overuse or misuse of an antibiotics, definitely it will create the drug-resistant superbugs. And now it's becoming a great challenge in both health and wealth economy. And how does antibacterial resistance happens with antibiotics? If you see here, there are a lot of antibiotics, and after we will take the antibiotics to kill the bacteria. By the mutation, the bacteria will get resist. Here I had given the orange color representation, which you treat as an antibacterial. And after I'm giving the antibiotics, which I am representing in a blue color, it is a resisted bacteria and rest of is the normal bacteria. After giving the antibiotics, the normal bacteria will get died, and the resist bacteria won't get died because of it shows resistant to the antibiotic. And these resistant superbug may increase their size and it will grow, and which will give resistance to the another bacteria. So this is the drug resistance of antibiotics with the antibiotic resistance. What is the current situation of superbug resistance to the antibiotics? So if you see there are a lot of antibiotics which will resist to the particular microorganisms or particular bacteria or virus. If you see, I had given a few examples here. If you're taking the antibiotic like fluoroquinolines, it will to treat the urinary tract infection, it resists to the E. coli microorganism. And one more example here. The treatment failure has been seen with the 3rd generation cephalosporins. If you are giving any 3rd generation cephalosporin drugs, antibiotics, the treatment failure will be seen with the gonorrhea disease. Gonorrhea treatment we will see the treatment failure. This is due to because of drug resistance. And this is happened in 10 countries. And this drug resistance, the treatment failure is happened in almost all 10 countries like Australia, Austria, Canada, France, Japan, Norway. Due to because of this drug resistance, the newly WHO guidelines had given that the quinoline drug is an antibiotic which should not be recommended to the gonorrhea because of it will spread the high risk of resistance. And antiviral drugs also, like if you see the amantadine and the rimantadine, these are the drugs of an antiviral category, which also shows the drug resistance to the all influenza virus. And if you see the methicillin drug, it will show resistance to the Staphylococcus aureus. And antiretroviral drugs, they also show the bacterial resistance. And how we count to minimize the superbug resistance to the antibiotics. Can we minimize them? Yes, definitely. By say small precautions, we can minimize. At least we can minimize the superbug resistance by taking antibiotics with the prescribed physician. After giving the prescribed antibiotics by the physician, then only you have to take the antibiotics, and never share the antibiotics or never use the leftover antibiotics. And do not use the antibiotics to the viral infection because the antibiotics are meant for bacteria, to kill the bacteria. It's not used to treat the viral infection. So we should avoid to use the antibiotics to treat the viral infection, and we have to wash the hands regularly, and we should be not close to the contact with the sick people. And what are the impacts of drug-resistant superbugs? If you see a lot of impacts with the drug-resistant superbugs, which may affect the physicians, patients, students, pharmaceutical producers, public, and each and everyone in the society. And because of this drug resistance, the cost of an antibiotic also getting increased. And for example, if you're given an antibiotic to kill the microorganism, when these microorganisms had showed the resistance to the particular antibiotic, the cost will be increased to 3 to 18 times. So the cost is also plays an important role in the drug resistance. And if you're having any severe conditions of drug-resistant superbugs, we have to stay prolonged time in the hospital. And recently studies, in a recent study, they had declared that 88 countries. In 88 countries, they had studied that the result had showing more than 54% patient having bacterial infection among the 54% of population who having the bacterial infection. Surprisingly, 70% of them had received the antibiotics. So the small survey had conducted in 88 countries. In that 88 countries, 54% of patients had proven the bacterial infection. Among the 54%, 70% of them had received the antibiotics. Either it may be prophylaxis or either to treat the disease. Treating the bacterial infection is not a big task, but yes, treating with the resistant bacteria, it may prolong the hospital stay also. And what are the problems facing antibiotic resistant superbugs in the fight against COVID-19? So the battle against COVID-19 had becoming increasing complex with two major phenomena. One is rise of secondary bacterial co-infection, and second one is SARS-CoV-2 mutation. There is one reported case here. When I'm searching the articles, very interesting point I had noticed is that one reported case, one woman who is in 70 years old. She had a bacterial infection after breaking her femur. She was taking the carbazepine antibiotic, which had showed the resistance, and these carbazepine resistance had shown rest of the 26 antibiotics also. And after 2 weeks, she met with a death also because of these multi-drug resistant antibiotics. So this is an important thing while taking an antibiotics. Even it is not advised to give the, even recently WHO had declared that you should not give any antibiotic unnecessarily unless whether the patient having the infection. Okay, matlab you should not give it in a prophylaxis, or should not give mild or moderate COVID-19 cases also. So the antibacterial resistance will increase with the high dose of an antibiotics, and which will be seen in the COVID-19 patient treatment also. Studies summarized that the clinical reports of 60 to 70% of COVID-19 patients had received antibiotics. So recently studies, they had declared this almost 60 to 90% they had received the antibiotics to treat the COVID-19, either maybe prophylaxis or treatment. So by this review article, I can conclude that by taking more antibiotics in COVID-19, antimicrobial resistance will get increased, and the toxicity also will increase because we have to increase the dose. And just now I had said that WHO guidance had said that we should not recommend any antibiotic therapy for prophylaxis patient to mild or moderate COVID-19 patient unless the patient having any symptoms of bacterial infection. By these bacterial resistance, the mortality also will get increased. The society have to understood that more confusing times of this COVID-19, antibiotics is not for is not meant for a better. Thank you.
Thank you, Bhavani. So your topic was current progress, impacts and challenges of drug-resistant superbugs on COVID-19 pandemic. Okay. So my first question is, how the drug resistance is being transferred from one microorganism to another? What is the mechanism of transferring this drug resistance?
Ma'am, if we see the resistance, it's of 4 types. Natural resistance भी होता है, acquired resistance, cross resistance, and multi-drug resistance होता है, ma'am. And when the bacteria itself naturally get resisted, and these natural resisted bacteria will transfer these resistance to the rest of the bacteria to survive with that particular antibiotic.
How? What is the mechanism of transfer? That is my question. How it transfers that resistance part?
Bhavani ma'am.
By mutations, ma'am.
Please, Bhavani ma'am.
Sorry to interrupt. One factor is responsible for that.
Asking for video. Bhavani ma'am, please on your video, please turn on your camera. Ask a question.
So what I told you, Mrs. Bhavani, that is one factor is responsible for this transfer. Okay. And second question is, what is the difference between third generation and first generation penicillin?
Penicillins, come on.
Penicillin or cephalosporin, whatever may be. What is the difference between third generation and first generation?
Ma'am, effects on the inhibiting of cell wall synthesis, ma'am. It had categorized the generations depending on inhibition of cell wall synthesis to the particular bacteria.
Okay. My third question is, in your topic, it was mainly about the superbugs on COVID-19 pandemic. Okay. But throughout you have described about the drug-resistant antibiotics. So, what is the effect of drug-resistant antibiotics on this virus, COVID-19?
Definitely, ma'am.
How it will affect?
Definitely, ma'am. If you are taking the antibiotics in a high dose, it may causes resistance. And now we are giving the antibiotics such as
so the bacteria will become resistant, I think.
Yes, ma'am.
Bacteria will become resistant. But how will it affect the virus?
मतलब मैम ये
Because
resistance how
this
it will affect the
antibiotics,
virus.
whatever we are using, azithromycin, etc., we are just combating the associated bacterial infections. Okay,
Yes, ma'am.
so I think drug
Even now in
resistant
clinical
superbug.
trials, even in now in clinical trials also they are not giving any erythromycin or chloroquine drugs to treat the COVID-19 because of antibiotics are not for the viral, ma'am. But now we are giving the antibiotics to minimize the symptoms. To minimize the symptoms, we are giving. For example, if the particular patient having the throat infection, we are giving the azithromycin
So
to minimize the symptoms.
I think it will not affect the COVID-19 pandemic. Means according to my opinion, it will not affect the COVID-19 pandemic. Okay. Okay. Thank you. Thank you so much.
I agree with Rupa ma'am. Like in case of viral disease, any viral, this antibiotics will not affect. But one question is another question, like how do you relate it to superbug? How can you relate COVID-19 with superbug? Like your topic, both the words like superbug and COVID-19 was not related to each other. Moreover, they're not like COVID-19 is nowhere reflected in your presentation. So how can you relate your topic with your presentation?
Ma'am, the superbugs will show it's in strains of bacteria and virus, ma'am. The superbugs is not only the strain of bacteria, it's a strain of bacteria,
but you're talking
virus,
in
and fungus.
I'm getting it. But you're talking in respect to COVID-19 as per your topic.
Yes, ma'am. Yes.
Then how do you mention that COVID-19 is a drug resistant?
Ma'am, definitely we are giving a prophylaxis, so we are treating with an antibiotic. Antibiotic, so maybe it may also affect the somewhat. It plays a role on virus also.
It is just associated organisms are being affected, and it is also a trial which is going on. It is still in the trial phase.
Profile phase, ma'am.
My suggestion is to please review your work. Please review your work. Thank you.
Sure, ma'am. Sure, ma'am.
Yes, Bhavani ma'am, I have one question.
Sir?
What are the current situation of the superbugs resistance of any one highly used antibiotic in the COVID-19 pandemic?
Sir, one more time, sir, question.
My question is, what is the current situation of the superbug resistance of any one highly used antibiotic? As you are saying that you have given some example of the antibiotics used in. So the antibiotics, what are the current situation of the superbug resistance of any one highly used antibiotics used in the pandemic situation?
So in pandemic, we are mostly giving the two antibiotics like azithromycin and chloroquine. The resistance will happen to which drug we are prescribing to the particular patient. And the symptoms are different from person to person, and the treatment of the antibiotics we are giving to the person is different. And so
okay, ma'am. Thank you.
Thank you, sir.
Thank you, Mrs. Bhavani Pentela. Now I would like to the next presenter, Mr. Karthik Singhal.
Hello.
Karthik?
Hello. Ma'am, I am audible?
Yes, you are audible. Clearly audible. Start, Karthik.
Yes. Ma'am, my slide is visible or not?
Yes, it is visible.
Yes, it's visible, visible. Yeah.
Slide show kar do, Karthik.
Ma'am, I'm already sharing my slide.
Yes, it's okay. No.
Good afternoon to one and all present here. My name is Karthik Singhal and I am from MPharm Pharmaceutical Chemistry, Department of Pharmaceutical Chemistry, Noida Institute of Engineering and Technology. Today, for presentation, my topic is implementation and application of AI to overcome antibiotic resistance. This is the content of my presentation. So, we first start from the introduction. In early times, many of the people died not because of cancer or tuberculosis, they died because of infections. Infections which they called blood poisoning because there was no method of treatment available at that time. And if they are lucky, they recovered, and if they are not, they died. And even a small scratch is the cause of their death. But the people dying because of infection again, because of phenomena called antibiotic resistance. And we play a game with a resistance. We made a antibiotic, bacteria made a resistance so quickly, and we again made an antibiotic, bacteria again made a resistance. But now the game is over because pharmaceutical companies are not interested in making new antibiotics by conventional methods. They try to find some new methods because they are very expensive and resistance occurred so quickly. As we shown here, penicillin introduced in 1943 and resistance starts showing in 1945. Vancomycin comes in 1972 and resistance start in 1988. Imipam comes in 1985 and resistance start in 1998. And daptomycin, which comes in 2003 and resistance start in 2004. So we have to change our approach, and our new approach is AI. And what are the applications of antibiotic resistance? What are the applications of artificial intelligence in a field of antibiotic resistance? First, prediction of antibiotic resistance. For the prediction of antibiotic resistance, we have software called FAST. For the antibiotic resistance infections and rational use of antibiotics, for this we also have software called Mycin and Gabot. Mycin is a software which used by CDC, US CDC. Antimicrobial peptides and antimicrobial combination. For this, we have software called Figil. So we look at here some common algorithm used for antibiotic resistance. First, we have Navy bias. This is a classifier and it is based on the bias algorithm. It gives a independent assumption. I also show here a research which is done by Rezi Hechusu, in which they develop a surveillance system for the hospitals, the resistance which is in the hospitals. The next is decision tree. Decision tree is a flowchart like structure in which we put a different data sets and it gives outcome in form of branches. It gives different decisions. So we analyze them and we work them accordingly. And Reynolds method. It is also from a research article. Uh, Reynolds developed a method to determine the cost which is used, which is used to which is used to find new antibiotics. Next is random forest. Random forest are the machine learning software, and it is also used for classification, regression, and and it is used for decision making. And here are Chandrasekhan model. Chandrasekhan made a Indus model, which is based on the random forest, Indigo model, sorry, which is based on the random forest, in which he used E. coli and tuberculosis stains, and found their compound uh, which are effective towards these stains. Next, we have support vector machine. In machine learning, support vector machine in machine learning, support vector machines are supervised learning models with associate learning algorithm that analyze data used for classification and regression analysis. The support vector machine, we put here are different data sets, and it analyze that data and give the amount and how we use our antibiotics, how much quantity, and how the ways which we use it. Next is artificial neural network. Artificial neural network is a component of artificial intelligence. uh, it is brain, work like human brain. It also makes a decision for us, so we can find a good output. And we here have a stroke study. Strokes give a model and predict antibiotic based on their structure by using the artificial neural network. And he created a zinc 15 database for after their study. So we have some state of the art antimicrobial support system. These all are softwares. uh, the Mercurio system, it is used for nosocomial infection, to determine the nosocomial infection in hospitals in Italy. It is used in Italy. Then we have gram vector. It is used by US CDC, and it is recommended by US CDC in America and as well as other countries also. Then we have Vonet. It is used for antibacterial susceptibility testing. And then we have TheraTrack, which is used for support pharmacy. So that there are proper, we can maintain proper record of the antibiotics. And we have also a research of Dr. Jim Collins. Jim Collins uh, is a bioengineer. He is working in a university, San Diego. Uh, he made a model to develop a new antibiotics, and he told that he make a 7 new classes of antibiotics in 7 years, upcoming years. As we know, we there is no new class of antibiotics from last 30 years. So TED funding organization fund them for that, and he also used their AI based system for development of new COVID medicines. And I end my presentation with Alexander Fleming's word. He said that while he receiving his Nobel Prize, the thoughtless person playing with penicillin treatment is morally responsible for the death of man who scrambles to infection with the penicillin resistant organism. I hope this evil can be averted. And here's my conclusion. We know the area of healthcare and the area of data sciences is very vast. And if we combine these two areas, we definitely found something new which is helpful in overcoming the upcoming next pandemic of antibiotic resistance, maybe. Thank you.
Thank you, Karthik. Hello.
Yes, ma'am.
Actually, it's a very good presentation, and I think very modern thinking, modern presentation. And we have also come to know a lot many things from your presentation. But in the earlier slides, you have mentioned it is this drug resistance, that game of antibiotic resistance is over. No, no, it's not yet over. Okay, but this application of these things you have described very nicely, this naive Bayes algorithm, decision tree, etc., etc. But you should have cited few more means practical based examples of antibiotics, okay, on which this AI application is already been there.
Yes, ma'am.
Otherwise,
Yes,
your presentation is very nice.
Karthik, please turn on your camera.
Sir, I'm trying, but it's not.
You can.
When you are replying to the questions, please turn on your camera. First, turn on your camera, then you can
Sir,
reply.
You remove your slide sharing, it will automatically. Okay, okay, thank you.
Yeah.
Now it's okay.
Thank you, Karthik. Very nice presentation. Just 2 points I have mentioned there. Okay.
Okay, thank you, ma'am.
No questions from my side, Karthik. The presentation was nice. Thank you.
Thank you, ma'am.
Karthik, I have a question.
Yes, sir.
The first question is, can you give the at least one application of Razel-Hashin model and the Chandrasekhar model as you have described in your presentation?
Sir, Chandrasekharan develop a model, Indigo model. It gives a prediction of antibiotics. Basically, चंद्रशेखरन ने ई कोलाई और ट्यूबरक्लोसिस के बैक्टीरिया लिए और उन्होंने मल्टीपल जो लिगेंड्स थे उनसे उनकी स्टडी करी, मल्टीपल लिगेंड्स की, और फिर उन्होंने कुछ ऐसे कंपाउंड्स आइडेंटिफाई करे जो पोटेंशियली ऐसे कंपाउंड्स हैं जो कि रेजिस्टेंस डेवलप नहीं करेंगे इन फ्यूचर. उनका मॉडल ये था.
Okay, very good. And next question. And you have mentioned that halicin. You have given one example, halicin.
Yes,
and
sir,
it is
halicin.
predicted as an antibacterial molecule used in drug repurposing.
Yes, sir.
So can you elaborate on how halicin is predicted as antibacterial molecule used in drug repurposing?
Sir, actually, halicin is recently finded molecule. In sorry.
Okay, thank you, Karthik.
Welcome.
Thank you, Karthik, for your nice presentation, and thank you, the judges. Next, we are calling Divya Sharma as the presenter. Miss Divya Sharma, you can start your presentation.
Thank you, ma'am. Am I audible?
Yes,
hello, Dr. Sanjita. Dr. Sanjita, you just mentioned the name of the institution also.
Okay, okay.
Okay.
Okay, ma'am. Sure.
Thank you.
Divya Sharma, she is working as assistant professor at NIT Pharmacy Institute, Greater Noida, and she is going to present her oral presentation. So, Divya Sharma, you can start your presentation. Please keep your camera on. Okay, thank you. And make it slides.
Sorry, ma'am, but I'm not able to present my PPT. My screen is visible.
Yes, ma'am, it is visible.
Okay, so good afternoon all. Firstly, I would like to thank Dr. Vijay Majumdar sir, Director of Pharmacy of Institute NIT, and for giving me this opportunity to present my topic on antibiotic resistance, main types and the causes. So let's discuss about the antibiotic resistance. What exactly is antibiotic resistance? So antibiotics are the drugs that are used to treat the infectious diseases or the diseases caused by the microorganisms. And antibiotic resistance is a kind of a process of bacteria not being able to kill by the antibiotic medications or the drugs. So bacteria are having the capacity to resist the antibiotic drugs. Let's discuss how it all gonna happen and how bacteria becomes resistant to the antibiotics. Well, antibiotic resistance is a kind of a natural phenomena where the best bacteria can. Hello, ma'am, my voice is audible.
It's audible. You can go. You can carry on.
Okay.
Yeah, continue. Yeah.
Okay. So antibiotic resistance is a kind of a natural phenomena. As you can see in the picture, where the best bacteria can survive and the weakened one die out. And antibiotic usually doesn't get all of the bacteria. Some bacteria survive and they reproduce further and further. And all of the bacteria, survived bacteria, become resistant to the antibiotic. Now, as I said earlier, that it is a natural phenomena, but there are certain activities, if I precisely say, the human activities that enhances or accelerated this problem, antibiotic resistance. For example, if we overuse the antibiotics, if we misuse the antibiotics, and if we are taking the inappropriate dose of the antibiotics, so this may enhances this problem. Now, what are the different types of the antibiotic resistance? Generally, we have 4 different types: the natural resistance, acquired resistance, cross resistance, and multi-drug resistance. In natural resistance, in this, the bacteria have the ability to resist these antibiotic medications by reducing the permeability of the outer membrane. For example, gram-negative bacteria is resistant to the vancomycin. In case of the acquired resistance, there is some genetic changes in the microorganisms happen. For example, if there's a mutation or changes in the gene expressions, then it causes the acquired resistance. For example, E. coli, due to mutations, it resists the trimethoprim antibiotic, and certain fluoroquinolones drugs shows the acquired resistance. The third resistance is cross resistance. When a single resistance mechanism confers the resistance to the entire family of, entire class of the antibiotics, then it is known as cross resistance. For example, if Staphylococcus aureus is resistant to, shows resistance to erythromycin, then it also shows the resistance to the neomycin and kanamycin. Last resistance is a multi-drug resistance. It is the most dangerous one also, because in this, the microorganisms resist different drugs of the different classes. For example, in this example, the Staphylococcus aureus is not only resist the methicillin, but it also resists the aminoglycosides, macrolides, tetracyclines, and chlorine. The another example is in case of tuberculosis. Mycobacterium tuberculosis is able to resist the isoniazid and rifampicin also. So these are the types of antimicrobial resistance. Now, why does this problem exist? So one of the main reasons of this problem is because antibiotics are often being overprescribed and are being given to the patients who are not necessarily need them. So according to this report, CDC Threat Report 2013, in this report, 50% of all antibiotics prescribed are not needed and not necessarily need. Even in the ICUs, in intensive care units, 30 to 60% of antibiotics are not necessarily needed, but still they are prescribed. Another major reason of the antibiotic resistance is the agriculture use, extensive agriculture use. In this, the healthy animals or the livestock are given antibiotics to prevent the diseases and to promote their normal growth. But they received antibiotics, so the resistant bacteria develops in their guts. So the milk products or the meats or the products that are obtained from the animals is eaten by the humans, and then humans have those antibiotic resistant bacteria. For example, tetracycline and streptomycin, they are sprayed onto the fruit trees and in the US. The reasons are inadequate sanitation and inappropriate course. Like if patients are not completing the full antibiotic course, then the antibiotic resistance may be enhanced. Other than that, lack of discovery of new antibiotics is also one of the major cause of antibiotic resistance. So how can we tackle this problem? WHO launched various programs, and recently in 2019, they launched the AWARE program, in which they classified different antibiotics in 3 categories, that is Access, Watch, and Reserve. In the Access category, they classify the antibiotics, those antibiotics that have activity against the wide range of the susceptible pathogens or bacteria, and they have some lower resistance potentials. In this Access category, approximately 48 antibiotics included, out of which 19 are individually added into the WHO model list of the essential medicines. Similarly, in watch categories, there are certain antibiotics included, and similarly into the reserve category also. Reserve category is treated as the last resort option or last options which should be accessible, but they should be only used when there are no options left or there are no alternatives left. In this, 22 antibiotics are included, out of which 7 is included into the WHO model list. WHO also launched certain newer drugs. For example, these 3 drugs targets the multi-drug resistance and categorized under the reserve category. Certain other WHO initiatives to address this problem, this antimicrobial resistance problems, for example, WAR, World Antimicrobial Awareness Week. This program is held on 18 to 24 November of every year, and its the main aim to aware the public or health workers or the policymakers to avoid the spread of drug resistance, and similarly GLASS, GARDP and IACG as well. Now, what to do? How can we reduce this antimicrobial resistance? Generally, we have certain parameters by which we can reduces this problem. Firstly, we can set up some rules and regulations to some rules and laws to set up the usage of certain of these antibiotics, and of course increasing the investment in R&D for the new antibiotics so that we stay ahead from the resistant strains and improve the access to high quality antibiotics, and prohibit the use of antibiotics for the healthy animals. We can use these antibiotics onto the sick animals, but I think we should not use these antibiotics onto the healthy animals for their normal growth and normal disease resistance. And future perspective. I think, if I'm not wrong, I can say that antibiotic resistance is a silent pandemic, and it is a major threat to the global health. And it will take a huge effort by the research scientists or the policymakers or the individuals, but by all together we can make a difference. Thank you. This is all about the antibiotic resistance main causes and time.
Thank you, Ms. Divya. So I have three questions to ask. First of all, number one, what is the name the organelle which is responsible for transferring the drug resistance? Okay. Number two, that is what is the difference between cross resistance and multi-drug resistance? And number three, what is the reason behind cross resistance? First of all, name of the organelle which is responsible for transferring the drug resistance.
Sorry, ma'am.
Divya,
I didn't
remove
answer
your
the
slide
first
share.
question. I can.
Please make your camera on.
My second question is difference between cross and multi-drug resistance. And third question is what is the reason behind cross resistance?
Ma'am, can you see my picture?
No, you are not visible yet. No, it is not visible now.
And now?
No.
No, it is not visible.
Make your camera on. Come out of slideshow and now.
Put off your that part, slide sharing.
Now, ma'am? Now I'm.
Yeah. It's okay. Please answer ma'am's question.
Yes, ma'am. So I'm not able to answer your first questions. Can I answer your second and third question, ma'am?
Yeah, of course.
Okay. So your question is what is the
cross and
difference between cross resistance? Yes.
Multi-drug.
Ma'am, in cross resistance, yes, ma'am. In cross resistance, when a microorganism is resistant to one, if we are taking ofloxacin and I am resistant to the ofloxacin, and if I'm again taking the ciprofloxacin, then my body resists to the ciprofloxacin also because it is of the same category drug. And then your
so this is called cross resistance. Okay.
Yes, ma'am. This is called cross resistance. In case of multi-drug resistance, then there is a resistance between two different drugs of the different categories. Then it is known as the multi-drug resistance. For example, in case of, I had given the example of Mycobacterium tuberculosis. In case of TB, the Mycobacterium tuberculosis also resists the rifampicin and the isoniazid. So to the different classes, to the different drugs. This is called multi-drug resistance.
Okay, thank you. And this that third question was, what is the possible mechanism for developing cross resistance?
Mechanism.
Mechanism or reason?
Yes, ma'am. Ma'am, it may be because if a certain drug is treating the because the pathology of a drug causing the. Can I speak in Hindi? Suppose, ma'am. Okay, ma'am. मैम अगर एक ड्रग जो है वो सेम मैकेनिज्म से किसी सेम पैथोलॉजी जो है पैथोफिजियोलॉजी शो करके एक डिजीज कॉज कर रही है, तो अगर हम उस मैकेनिज्म को ही ड्रग से इनहिबिट कर देंगे तो वो उसकी वजह से जो है वो क्रॉस रेजिस्टेंस आ सकता है।
नहीं मैम, नहीं। चलो नेक्स्ट जज होगा। थैंक यू दिव्या, थैंक यू।
ओके। हेलो दिव्या। Yes, sir. Salahuddin sir, yes.
Yeah, I have one question.
Yes, sir.
You have mentioned plasmomycin in the AWARE program launched by WHO. So can you categorize And what are the possible mechanism of action of this medicine?
Sorry, sir, can you repeat the question?
Plasmamycin. You have mentioned in the one program launched by WHO, in the AWARE program, you have mentioned one newer drug, plasmamycin. So, can you categorize this medicine or and the mechanism of action of this treatment?
These all three drugs are categorized under the reserve category, sir. In the reserve category of the AWARE program.
And the mechanism?
Mechanism, sir, I'm sorry.
Okay. Okay, Divya. Rashmi, please ask.
My question is, what is the mechanism for antibiotic resistance?
Ma'am, it is due to the genetic expression, changes in the genetic expression. It may be due to the mutations, and naturally some organisms can cause resistance naturally. They shows the natural resistance, and there is a mechanism of HGT, horizontal gene transfer.
Now the second question is, will it pass to next generation? And if it does, then how can it be resisted, passing to next generation?
Can you repeat the question now?
The ability of resisting a drug, does it pass to next generation of the organism? I mean, is it transferred genetically? And if it does transfer, then how can it be resisted? How can this phenomena be broken of transferring drug resistance in next generation?
Yes, it is passed through the next generation, but actually I exactly don't know what you asking.
If like it is passing from one generation to another in case of bacteria, right? Drug resistance ability. Now, if I want to break it, if I want to change it, then what will you do? What is the pathway that is followed?
Means you want to reverse the antimicrobial resistance. You asked for this. Yes, ma'am. We can. Yes, ma'am. We can reverse this process of antimicrobial resistance. But I have not gone into the detail, so I cannot explain this further. But yes,
no issues. Thank you.
But my answer is
no
yes.
issues. Thank
Okay. Okay, Miss Divya, your presentation was good, and you faced all the questions and tried for answering the questions. Okay, thank you, Miss Divya. Next is Miss Richa Sakia, who is working as assistant professor at NIT Pharmacy Institute. Please, Richa Sakia, you can present your oral presentation. You can start your presentation.
Good afternoon, ma'am. I'm audible?
Yes. Please start your
Okay.
presentation.
Good afternoon, director sir. Good afternoon, judges, and good afternoon to all the participants. And I sincerely thanks to the director sir who gave me this great opportunity to express my views on the topic of antibiotic drug resistance and causes and their future challenges. So I will start my presentation. Ma'am, is my screen visible?
Visible.
Okay, ma'am. Thank you.
Just make it slideshow.
Yeah, sure, ma'am. So, my topic is on the antibiotic resistance causes and the future challenges in the digital India. Nowadays, most of the persons are taking the antibiotics without any prescription, either if the patient is suffering from simple cough and cold or either fever, they directly take the antibiotic without any prescription. So this may the cause. This is the one cause of the antibiotic resistance. So I will start my presentation. So in during my presentation, I include the introduction about antibiotic resistance, etiology of the antibiotic resistance, mechanism of the antibiotics, and after that I will discuss about the mechanism of the antibiotic resistance, prevention of antibiotic resistance, and future challenges for the antibiotic resistance. So what is an antibiotic? First of all, antibiotics are the drugs which are able to kill or which are able to static the growth of the bacteria. So antibiotics have the two types of characteristics. One is your bactericidal and one is your bacteriostatic. Bactericidal means when the drug is able to kill the bacteria. And what do you mean by the bacteriostatic? When the antibiotic are able to stop the growth and the replication or the reproduction of the bacteria reproduction. Okay, so the antibiotic resistance occurs when the germs like bacteria and fungi develop the ability to defeat the drugs designed to kill due to them, which mean that the germs are not killed out. So it continuously grow. So this is the antibiotic resistance. So the infection caused by the antibiotic resistance germs are difficult and sometimes it's impossible to treat. So the antibiotic does not mean that the body is becoming the antibiotic resistance. It is just like a bacteria have a capability to resist that antibiotic designed. Okay. So what is the etiology behind the antibiotic resistance? So the etiology behind the antibiotic resistance is to increase the use of antibiotics. Prescription not taking properly means if the doctor or if the physician recommended that take a medicine in the morning or in the evening or in a BD or a QD, so the patient most of time the patient are skip their doses. So it is also a cause of the some type of drug resistance. So without any medical supervision. So it is a major cause because the antibiotic is like a OCD. It's the over-counter drug. Antibiotic used for the viral infections and the spread of the resistance microbes in the hospital due to the lack of hygiene. Patients who do not have complete the course, as I earlier discussed, that the patient is not taking the medications properly. And next is the antibiotic in the animal feeds. Next, drainage of the antibiotic in river or in the water bodies. Sometimes ago there is cases in the Yamuna river that the amount of the antibiotic is too high in the river water of the Yamuna. So this is the also cause of the antibiotic resistance and inappropriate way of discarding of the antibiotic medicines. Sometimes what the people they do, if the medicines are expired or if the medicines are not in use, what they do, they just dump in improper way. A major cause of the drug resistance. So how it is spread means how the antibiotic resistance is spread. So if the physician or a doctor gives a treatment or gives a prescription to the uh, patient to take these antibiotics, so the patient take their antibiotics in an improper way, it not follow the proper prescription, so that it becomes antibiotic resistant. So after that he or she will go to a hospital or in any public area with an unhygienic condition. So when he or she will be communicated or coming in the contact with a healthy person, so it will also infect it to the another person. And the other cause of the antibiotic resistance to spread is the antibiotic dose are given to the animal food, so or in a crops, so that the animal we take the dietary products. So at that time we are also taking that antibiotic drugs which will be given to the animals or in a crops. So we will consume that food. So again, we are uh, antibiotic drug resistance patients. So what is the mechanism of action of the antibiotics? So basically, there are some steps by which the antibiotics can do a actions of their actions of their effects or to the bacteria for their stop of their growth, something like that. So the first mechanism of action is just like to inhibit the cell wall synthesis. If the cell wall is not properly synthesized, or a drug which is we taking the mode of action of a drug that is to inhibit the cell wall synthesis. The example of that drugs are the beta-lactam, just like penicillin, cephalosporin, carbapenems, okay, or vancomycin. And the another category of the drug which are inhibit the DNA gyrase, means it inhibit or it breaks the DNA molecules, so that the DNA molecule is not properly produced or properly built up, so that the DNA is not built up, so the bacteria cannot survive. So it's a effect of the bactericidal effect. So the bactericidal drugs are like a quinolines. Some bacteria are inhibit the RNA polymerase synthesis, just like a rifampicin. And there are some drugs which are inhibit the protein synthesis. Okay, in bacteria there are the two subunits of the bacteria. Sorry, this ribosome, that is a 30S and the 50S. Okay, so the 30S, the 30S inhibitors drugs are like a tetracyclines, aminoglycosides, and the 50S subunits inhibitors are like macrolides, clindamycin, chloramphenicol. These all are the drugs which are inhibit the protein synthesis, so that the bacteria cannot produce a proper amount of protein. So they are bacteriostatic. So the proper amount, the proper growth of the bacteria does not occur. And the another category is the folate synthesis. Means in this the inhibitors are the inhibitors acts on the PABA. These are the PABA inhibitors. Folate synthesis of PABA inhibitors. So when the PABA inhibitors are inhibited, so the DHF, that is a dihydrofolate, it is not converted into the tetrahydrofolate A. So this process is stopped by the help of the some drugs like sulfonamide, trimethoprim, cotrimoxazole. These are the drugs which are used for the inhibitors of the folate synthesis. So I will discuss next. I will discuss about the what is the mechanism behind the antibiotic drug resistance. Means so there are the some points. There are the some steps by which the drugs can register the antibiotic resistance can occur. Just first step is the antibiotic drugs. Sorry, first is the inactivation of drug. So inactivation of drug, what happened? There's a two cases, just like either a inactivation of drugs or a modification of drugs. Right. So in the inactivation of drugs, what happened? The bacteria are inactivate the drugs by the help of the resistance genes. So what is the resistance genes? Resistance genes are the resistances. What they do? They make the antibiotic counteracting site, just like a beta lactamase. So the beta lactamase when activated, so it activates it inactivates the penicillin G. So the penicillin cannot affect just like a proper way. Okay. Then the next mechanism behind the
Excuse me, Richa. Excuse me.
yes, ma'am.
Richa, there is some time bound. So please wind up your presentation. Okay?
Okay, sure,
Okay.
sure, ma'am.
You
So
can
I will
continue.
find a few
Yeah,
minutes.
you can continue.
So okay, ma'am. So next is the alteration of the protein enzyme receptors. Just like every site, every sites have a specific protein receptor binding site. So in the antibiotic resistance, what they happen, the targeting sites or the receptor sites are altered. So the exact location of the binding site or drug are not allocated, so they are not affected as well. Next is the activation of the drug efflux. So drug efflux means when the uh, uh, when the efflux pumps are removed. Okay, when the efflux pumps are removed, so the entry of that antibiotics are not properly go to the cell, so the bacteria are not killed. So these are the points by which the mechanism of action of the antibiotic resistance. So there are the two types of important genetic mechanism of antibiotic resistance. First is mutation. So how the mutation take place? So it is caused. It is take place by the resistance gene. So what the resistance genes do? They are two type of vertical gene transfer or a horizontal gene transfer. After that, a second point is the active site of a new gene material means a gene which is already modified. It is transferred to the one generation to the another generation. So this is also a main cause of the antibiotic resistance. So the global plan of action of the antibiotic resistance are the 5 studies, just like to improve the awareness and understanding of the antimicrobial resistance, to strengthen the surveillance and the research, to reduce the incidences of the infections, complete the proper treatment of the antibiotics. So there are some core steroids means there are some steps to how to overcome from the this antibiotic resistance. So educate properly, educate the staff. There is some surveillance of the antibiotic resistance. So prescribing the appropriate amount of the antibiotics to the to the specific disease. Okay, so these are the some words. So the future challenges for the antibiotic resistance are to enlist the specific antibiotics, so that when we buy, first of all, already we have to purchase or we have to buy antibiotic with the help of the any prescription. Without prescription, the medical store person will not give the antibiotics to the person, and this is again the same thing. After that, the next thing is the collecting the data from the antibiotic resistance means which drug is most cause the antibiotic drug resistance, yeah, or in which area the drug resistance cause mostly. Stop the abusing of antibiotics in agriculture, stop abusing of antibiotics in humans. And after that, the ramp up of the infection prevention means the ramp of this infection prevention means to start the awareness of the how to provide a hygienic condition, properly hand washing, proper sanitization. All the conditions are from all the conditions of the person are well aware. So thank you.
Thank you, Richa. I have 4 basic questions for you. First, number 1 question is that in your topic you have mentioned that is future challenges in digital India. But throughout your presentation, I haven't found anything which is correlating the antibiotic resistance to future challenges in digital India. So what do you mean by digital India and how can you correlate your presentation with that? This is my first question. Can you answer?
Okay, ma'am. Okay, ma'am. Ma'am, just like no, digital India, just like the present scenario is based on the AI. Is the present scenario is based on the
Yeah,
AI?
but you haven't mentioned anything about that. But you haven't mentioned means a single slide also on this part, digital India. Achha, number 2 question, that is the probable mechanism of bactericidal and bacteriostatic activity.
Rupa ma'am, sorry for interruption. First, I'm requesting you, Richa, please turn on your camera.
Yeah.
Sure, sir. Sir, I will try to start the camera, but I'm not able
ऑफ
to
योर
start
स्लाइड्स ऑफ कर दो, कैमरा ऑन हो जाएगा।
Okay,
हाँ,
fine,
देखो फिर कैमरा का जो साइन है उसमें क्लिक करो, ऑन हो जाएगा।
Ma'am, I'm visible right now?
Yeah.
Yes.
Okay. So my second question is mechanisms of bacteriostatic and bactericidal activity.
Okay, ma'am. Bacteriostatic means we just try to
I know
inhibit
the meaning.
proteins.
Just mechanism. Yeah.
Pardon, ma'am.
Acha. Third question. My third question is, who is actually developing the resistance? The human being and the animals, the bacteria, or the antibiotic?
Ma'am, sorry, please repeat the question.
Who is developing the resistance? The human being and the animals, the bacteria, or the antibiotic?
It's by the antibiotic.
It's a very basic question.
Exactly.
Who is developing the resistance?
Ma'am, it's a bacteria. No? Actually, it's a resistance.
It's a bacteria.
Its capacity.
The bacteria becomes resistant. Achha, fourth question is, you told that these ribosomes, they are having 2 parts, 50S, 30S. What is the full form of ACE?
Some units.
S.
50 units.
That is, the unit is having a name, S. S becomes silent, S remains silent. That is Vedberg. Okay, thank you, Richard. Dr. Rashmi madam can continue.
Thank you, ma'am. Richa, how can you overcome the challenges of drug resistance? And another, this I'll mention that there was no correlation with the topic and the presentation. Question is just one, that how will you overcome the challenges? Richa, can you hear me?
Hello?
Richa, can you hear me?
Yeah, ma'am. Ma'am, actually, my laptop is currently not working. I don't know. So I will online with the different ID, that is Sonia ma'am. So I will audible. Like I'm.
No. Please be there. You are audible and visible. Just for 2 minutes it is there. So please be there.
Okay, ma'am. The laptop is not properly working. I don't know. It's hang up.
So can you hear Rasmi ma'am's question?
Yeah, ma'am.
Okay, Rasmi ma'am, please ask her again. She can answer.
How can you overcome the challenges of drug resistance bacteria?
First of all, we have to try to overcome from the abuse of the antibiotics. In the simple cases, what happened? if we're suffering from the simple cold or a viral infection, we just take the antibiotics. First of all, this thing, and second thing is that the basic concepts that it's only it's not on the OCT, means that the antibiotics never given to over the counter drugs, means it's always given by the specific prescription by the doctor or the physician.
Okay, thank you, Richa.
Richa, the same suggestion, which I'm giving you as the Rupa ma'am has given you the suggestion. You have to topic specific, and one more thing is time bound. Okay, I have given the time that is 10 plus 5 minutes. So you have already crossed your time limit. So I have one question regarding the future challenges. So what are the possible cases as a future challenges of the antibiotic resistance in the digital age?
Please you repeat the question.
What are the possible claims should be taken as the future challenges in digital India, and how it can differentiate from the current situation?
There is some disturbance. I don't know what you say. Hello.
Am I audible to you?
Right, sir. Right now audible.
My question is, what are the possible cases should be taken as the future challenges in the antibiotic resistance in the digital India? So, and how it can correlate with the current situation?
Okay, sir. Right now, some specific drug resistance, just like E. coli infections, in specific that patient take the antibiotics. So we will.
Richa, I'm talking about the future challenges.
Okay, so sir, in the digital India, we are trying to make the specific types of sensors, so that we detect the or we collect the data from which area or which kind of specific type of drug resistance will take place.
So, can you correlate the future challenges and the current situation?
Future challenges, sir, are that just like to actually in the digital India, just like if we talk about the digital India, so in the future challenges, like we are just move towards the technology, just like AI, just like ML, just a machine learning means we are just produce specific softwares. Right now, we just collect the data by the some prescriptions orders or something like that. So this is the future challenge from the current and the future scenario.
Thank you, Richa. Thank you.
Okay, sir. Thank you.
Thank you, Miss Richa. Your presentation was good, little prolonged one, but it is good. And thank you, judges. Next, I would like to request Vikas Sharma, NIT Pharmacy Institute, to present his oral presentation. You can start, Vikas, for your presentation.
Good evening, ma'am.
Good evening.
Am I audible?
Yes, you are audible. Just on your camera or your slide.
A very good evening to all the respected teachers, all the jury members, and all the participants present here. My name is Vikas Sharma. I am from MPharm first year, Pharmaceutical Chemistry branch from the NIT Pharmacy Institute, Greater Noida. My topic for the presentation is natural products and immunity as additional weapon against coronavirus replication.
Excuse me, Vikas. Can you show your slides, or your camera is off and slides are not visible? Your PPT is not visible. You can share your slide.
Ma'am, now visible?
No, it is not visible. You can share your slides.
I'm now visible.
Mm-hmm. No. Yes, it is coming. Just make it slideshow. Press F5 and start your presentation.
Hello. Hello.
Yes, please start your presentation.
Is it visible?
Yeah, it is visible.
Okay. My name is Vikas Sharma. I am from MPharm first year, Pharmaceutical Chemistry branch from NIT College Pharmacy Institute, Greater Noida. My topic for the presentation is natural products and immunity as additional weapon against coronavirus replication. So I want to start with the antibiotic resistance. In the simple meaning, antibiotic resistance means the inability of an antibiotic against the bacteria. So in the process of antibiotic resistance, the bacteria becomes developer resistant against the antibiotic. So to kill the bacteria, we require a higher dose or the alternative medicine, and it may prove the toxic effect. And the corresponding image tells that the antibiotic is small against the resistant bacteria, and resistant bacteria is playing with the antibiotic. Antibiotic is not affecting the bacteria, but bacteria is affecting the antibiotic in the opposite manner. So if I talk about the causes of antibiotic resistance, not only a single person is responsible for the antimicrobial resistance, but most of the human beings are responsible for it, including the doctors, pharmacists, patient, and the non-technical persons like farmer and even government are also responsible about the antibiotic resistance. World Health Organization also states that we are really facing a problem. WHO also states that we are facing a problem, and if we don't take action against it, then a dreadful post-antibiotic pandemic can start. Coronavirus. Nowadays, every person of the globe, every person of the earth is now aware of pandemic coronavirus because corresponding images are showing that coronavirus has putting a mask all over the earth. Coronavirus is a virus, is a RNA virus which belongs to the Coronaviridae family. And if we talk about the symptoms of the coronavirus, then it includes from the common cold to the severe respiratory diseases. But various strains of the coronavirus are also not showing any symptoms, but they are corona positive patients. Nowadays, coronavirus has just reached to more than 210 countries around the globe, and more than 2 crore positive patients are in the earth, and more than 8 lakh people have lost their life. Why? Because there are lack of synthetic inhibitors against coronavirus, and this is the biggest challenge. But the effectivity of herbal agents is a very good ray of hope. I would like to include an example from the Sushruta Samhita, that is a very respectable book about the pharmacy and the medicinal line. I would like to put a shlok from the Sushruta Samhita. Prashast desham sambhutam prashaste hani chodatam युक्त मात्रम मनिस्कांतम गंधवड़ रस अविंतम दोष धन मंगला निकार विकारी विपर्यय समीक्ष तंतम काले च भेषजम पाद्योच्यते। अर्थात उत्तम प्रदेश में उपजी हुई, उत्तम दिन में उखाड़ी गई, युक्त मात्रा में प्रयुक्त करने के लिए मन को शोभित करने वाली, गंध वर्ण रस से युक्त, समस्त दोषों का नाश करने वाली, विपरीत परिस्थिति में भी लाभकारी ऐसी कोई भी वस्तु जो रोगी का विकार दूर कर सके, उसको उत्तम औषधि कहा जाता है। Herbal products are the gift of God, which are always helpful to treat the patient situation in every disease and in different manners. Herbal medicines also having the immunobooster properties, and it plays the major role against the disease. This is the graphical abstract. First image is showing the coronavirus, pandemic virus. So how it is communicated into the host body, into the host body or from one body to another body. Basically, coronavirus replicates its structure by the help of few proteins like S protein Angiotensin convertase enzyme interaction, helicase protein, N protein, or PL-Pro etc. proteins. So for the when we search about the drugs against coronavirus, so we have to find out the inhibitory moieties. Against I have searched a few of the natural products which are showing the immunity properties of patient. These are the natural products. Example of tested natural products against the coronavirus replication. Number one, the quercetin derivatives or the beta galactosidase derivatives of quercetin. Name of the scientist Chen et al. They performed a study in the 2006. They showed that derivatives of quercetin inhibitory properties against 3CL Pro. And 3CL Pro is a protein that is showing the inhibitory activity against the coronavirus. And the extract of second substance is the extract of 121 traditional herbs. It also shows the inhibitory activity against spike protein. And the third drug is the Esetis indigotic. The extract of Esetis indigotic, it shows that it is also a potent inhibitor of the 3CL protein, and it is responsible for the replication of coronavirus into the host body. So the inhibition of these type of proteins which are responsible for the replication of coronavirus may be a proved natural substance that may have to overcome the coronavirus. And the aqueous extract of Houghtonia cordata were also investigated, and the extract of Houghtonia cordata shows the activity against 3CL Pro and RDRP protein. It was also found a better molecule having immunomodulatory properties. And the drugs named as Storia nucifera, Tribulus territris, Sambucus formosa, and the Angelica cassici. These drugs have also shown the activity against the enzymes, against the proteins like 3CL Pro, PL Pro, NL63. So, and these proteins are responsible for the replication of coronavirus in the host body. So the inhibition of these proteins, inhibition of the proteins which are responsible for replication of coronavirus. When we inhibit these proteins, then they can be a method to inhibit the activity of virus. When we come to the immunity concern, I would like to rename the immunity system as the protective system. The immunity system is a system which tracks the germ in the patient body. And the main part of the immune system are WBC, white blood cells, antibodies, lymphatic system, bone marrow, thymus, etc. Basically, WBC are the key players in our immunity system. They are made in our bone marrow and they go throughout the body. They look for the foreign microbes such as bacteria, fungi, etc. When they find any microbes in the body, they launch an immune attack. And when they launch the immune attack, the immune system execute the antibodies from the body and they fight against the antigen and the immunity takes place. Basically, few of the mostly students becomes confused when we talk about the immunity. So I would like to quote an example of mobile phone. Every person have the smartphones or the mobile phone in their hands or in their pockets. So for the safety of mobile phone, what we use the antivirus system. We use the antivirus apps. Why? Because to protect our mobile phones. So similarly, in the human body, God in the phone we install antivirus system and God installed the immunity system in our body. Similarly, in the mobile phones, we require the new and strong antivirus systems to show effectivity against the virus or trash media. And in the body also, immune system should be strong. Immune system should be strong against to showing the activity against microorganisms or the viruses. Sorry, ma'am, slide is not going forward.
And because your time is also exceeded, please see.
Okay, thank you,
Okay, ma'am.
Vikash. We
I
can
would like
ask
to
your
conclude
questions.
without the slide. I would like to conclude that I also have in the next slide the few examples of the herbs which show the good immunity and the anti-coronavirus activity. There are two enzymes, vitamin A retinol and vitamin C ascorbic acid, and the Corus calamus drug. These are the very good immunomodulators and showing the effectivity against the coronavirus proteins. And finally, I would like to conclude that review study suggests that coronavirus replicates itself by the help of few proteins that I named earlier in my presentation, and the natural moieties are inhibiting those proteins. So the inhibition of protein in the host body may deliver few of the new moieties that may produce new moieties against coronavirus. Thank you, ma'am.
Okay, thank you, Vikash. Acha, I would like to ask, not questions, I will like to tell you something. That is, though your presentation was very nice, your collection of data also was very rich, but there was some lack of correlation between the presentation and the theme of the e-conference. And in the title also, you were talking about natural products. Sometimes you talked about this antibiotic resistance and coronavirus replication. So the correlation I couldn't find throughout the presentation. Okay, so you should have focused on drug resistance and its challenges in digital India. So your presentation should have been theme based. That's much. Thank you.
Thank you, ma'am. I would like to request other judges also. Please ask.
Vikas,
yes, sir.
I want to ask you some question. Are you hear me, Vikas?
Yes, sir. Yes, sir.
First question. You have mentioned the two protein, this 3CL Pro and the PL Pro.
Yes, sir.
And some phytochemicals are obtained from some disease and active against these protein. So why these phytochemicals is active against only this 3CL Pro and the PL Pro?
Vikas, please come online.
Yes, ma'am, I am online. Sorry, sir. Hello.
Share your webcam.
Ma'am, camera is on.
You have not understood
visible.
my question?
Sir, please say once more.
Please switch off your PPT sharing.
Yes, sir.
Turn into switch on. Now it's okay.
Yes, sir.
So again, I am. You have mentioned the two protein structure, the protein that is 3CL Pro and the PL Pro, and these two protein is active against some phytochemicals which is obtained from some plants which you have mentioned in your presentation. So why these phytochemical obtained from this plant, obtained from this plant, active against the 3CL Pro and the PL Pro? Is there any specific requirement for the interaction of these proteins? No. So next question.
Yes, sir.
The basic question. SARS-CoV-1 and the SARS-CoV-2. What is the basic difference between the SARS-CoV-1 and the SARS coronavirus, SARS-CoV-2?
Sir, SARS-CoV-2 is also the also relates from the family of SARS-CoV-1, and there is minute structural changes. Sorry, sir.
Minute structural changes. I'm asking what is the difference between these two? Okay, Vikas, thank you.
Thank you, sir.
Like to invite our next Thank you, Vikas. Now I would like
Thank you.
now I would like to request our other oral presenter, Ashiru Anis Yau from Sharda University.
Hello.
Yes, Ashiru,
Good
you are
evening
audible.
to you all. Okay.
First of all, please turn on your camera.
Yeah, that's what I'm trying to do. Sorry, I'm trying to turn the camera on. I don't know if is the image clear, or is it still having a problem? Hello.
Yes.
Actually, my camera is turned on. I don't know if the image is if I'm showing on the screen.
In your screen, there is two things. One is camera, another one is your speaker.
Yeah, I have turned the camera on. The camera is actually
So
turned on
please
right now.
turn on your camera.
Yeah, it is turned on. It is turned on.
Turn on.
Okay, Ashiru, try to share your slide.
Yes. Sure.
Okay, Ashiru, you can continue.
Okay. Good afternoon to you all. My name is Ashra Anis. I'm a student of Sharda University, currently pursuing my MSc in Pharmaceutical Science with a specialization in drug design and development. It's like a privilege to be with you in this lovely evening. I'll be presenting on the a systematic review on antimicrobial photodynamic therapy of curcumin-loaded nanoparticles. As we all know, bacteria has possessed like a very great threat to our public health. At 2018, WHO terms antimicrobial resistance as one of the major threats to public health. But then, what is antimicrobial resistance? Antimicrobial resistance happens when microorganisms such as bacteria, fungi, viruses, and other parasites are immune, or like the drugs such as antibiotics cannot work on them. Such kind of phenomena is termed as a resistance. So in multiple resistance, meaning a bacteria can be susceptible to several antibiotics, being like it has several parts of which the antibiotic can target its activity, being cell wall synthesis, protein synthesis, penetration of the cell wall. If bacteria can develop more than a resistance to one or more antibiotics, that bacteria can term as multiple resistance. But then why is this multiple resistance a great concern to our global? New resistance have been developing by bacteria. They threaten the ability of common infectious diseases that are very common, like that can be treatable decades ago cannot be treatable anymore, which leads to the mortality rate. The 10 and 20 initiative, I know a lot of my professors and teachers here are much aware of it. It's an initiative that was governed by the Federal Food and Drug Administration, the US FDA, in the year 2000. This initiative has only one common goal: to produce at least 10 new antibiotics by the year 2020. That is from 2000 to 2020, to produce at least 10 new antibiotics with different mechanism of action. After several studies, after several trials, after several attempts, up till date, apart from linezolid, a drug that was approved to be used by 2005, although its patency was available in 1997, so it doesn't count in the new initiative by the US FDA, so it will not be included. There is no any new drug after spending $1,000 US. There is no any new drug that has been produced between 2000 to 2000. A lot of drugs are in the clinical trials as I am talking, but there is no any marketed new antibiotic with a different mechanism of action as those that we have initially before, of which I will on my subsequent slide I will take a look.
Ashiru, sorry to disturb you. Your slide is not proceeding. It is fixed in a single slide only.
It's not proceeding. Okay.
No.
Actually, on my second slide, I'm discussing about the 1020 initiative.
Okay.
Yeah. So what other way do we look at to target to combat this multiple resistance? We look at the photodynamic therapy, antimicrobial photodynamic therapy. What do we mean by antimicrobial photodynamic therapy? The word photodynamic is already self-explained. The use of a light or a light source to make a destructive mechanism, like to destroy a substance. This is not new, as it has been used in the treatment of periodontal diseases.
Ashiru, your slides are not moving.
Sorry, maybe I'm having a network problem from
Try
this
to
end.
check.
Yeah. I don't know if the slide is still moving.
Press enter key. Yes.
Now it's okay.
Am I audible, please?
You are audible.
Okay. So antimicrobial photodynamic. Okay, thank you so much. Antimicrobial photodynamic therapy, as I stated earlier, is not a new term. Photodynamic therapy has been used in quite ages for the period of almost 20 years ago in the fight of periodontal diseases. That is diseases that are affecting bacterial infections that are affecting most of the gum. But here in case of photodynamic therapy, it uses a sensible light to make a destructive to the bacteria, like direct sensitive light. So why not use the exact mechanism of this photodynamic therapy to fight other bacteria? Because we have natural products that studies has reported to have some certain antimicrobial effects. So using this light source by the use of color, this our natural drugs can also use as such example. Antimicrobial therapy of silica nanoparticles of curcumin has been reported against Pseudomonas aeruginosa and Staphylococcus aureus. There is a little brief about curcumin. Curcumin is something that we all have in our kitchen. It has been used for 1,000 years. As India even terms it as the golden drug due to its color and several uses that has been inapplicable today. So apart from curcumin being an antimicrobial therapy as already established, it has also used as a photosynthesizing agent. So this serves as a molecule to look at when we are developing a drug that will act as a photodynamic effect. Already explained that these photodynamics have the ability to sense color and the light source to make a destructive mechanism, thereby releasing oxygen via the ROS mechanism. But curcumin initially, I know it has a problem with bioavailability. Various methods have been used to improve its bioavailability, formulation. Just as of recent, I published a paper regarding this curcumin, of which I will provide its link if someone can read for the for curcumin that has been used in nano formulations of availability of curcumin. So, in a brief, antimicrobial resistance over the generations. Antibiotic has been in development since 1930, and as I said, not by the 2005. But take a look at the time gap at which this resistance is being developed. By 1940, just 10 years apart to 1950, sulfonamide has been resistance has been observed, has been reported. Severally, antibiotics, a lot of reports have been reported about their antimicrobial resistance. So you see, this photodynamics will convert as an alternate therapy to this resistance that we are. It will not cover all, but it's an adjuvant. It's like an add-on for us to hope on while we are still fighting the resistance, while we are still developing new drugs. So I will go ahead and look at the already marketed or already study that has been used with the development of photodynamic therapy of curcumin. In a combatant biofilm for periodontal, this curcumin has proved and has provided a positive result as in the treatment of ventilator-associated pneumonia and chronic. This is like an in vitro study. This bacteria have been cultured on a biofilm, and then they have been injected with a curcumin photodynamic. A curcumin as the photosynthesizing agent, a source of light, and it shows a remarkable destruction of those bacteria. So this can serve as a purpose, as a reason for this therapy to be used in the destructive of wound healing, superficial wound healing, and furthermore, we are looking at the targeted therapy, so such as like already Helicobacter pylori in the treatment of ulcer. Curcumin is reported to have an activity of that. So using photodynamic therapy, we can also reduce the resistance developed by Helicobacter pylori. Again, the viability and evitability of dental caries has also been reported. Staphylococcus bacteria, the activity, we know it's like one of the most resistant bacteria right now. But then a novel curcumin noted the nanoparticles injected with the novel photodynamic therapy has also increased reduced the ability of this the resistance of the Staphylococcus bacteria. Conclusively, as already discussed, I'm running out of time. Curcumin-mediated photodynamics reduce variability of microorganisms, as stated a lot. Despite this, we are still battling with the availability of curcumin. We are still fighting with the sources of life. We are still fighting with this antimicrobial resistance. But I think it's time we take a look at another angle. A development of new antibiotics is not an easy work to do. It's something that takes years to. Even we can take a look at antibiotics that have the same mechanism of action. We just need to alter its 1 or 2 positions. It takes us years to develop. With the emerging of new technology to reduce the time for drug development, till date, there is no new antibiotic with a different mechanism of action that has already been studied. So it's time to look at technology at our disposal to fight these microbial infections that we have. Thank you.
Thank you. Your name is fun. That is Ashiru. Thank you, Ashiru. So a very good and modern presentation, and it is related to the theme of the e-conference also, that is the challenges in digital India. Now my question is that in your explanation, you have used photodynamic word. So photo, yeah. I understand, the mechanism also I have understood. So, but what is the why the word dynamic is used here?
Like that is the change. The dynamic is a word. the change. We are using a light. It will change
So,
part of the destructive that happens. It changes the color is more sensitive to the light. For me, using a golden yellow, that dynamic, it changes the properties of that yellow and utilize it to our purpose by producing the oxidative oxygen to make it destructive to our bacteria. That is the word dynamic that is attached to it.
Achha. So now tell me about the source of light. What type of light is used here, and what is the wavelength of that light?
If you look at my slide, my previous slide, we use a low energy LED light. And generally, a low energy LED light can be
low energy LED light.
Yeah, the more advanced light presently, even it's the LED. Our technology.
Okay, and what is the source? Means it is an external source of light. How will you apply the light for this therapy?
Okay, the light has to be cultivated in such a way that the presence of that color is there are different types of color and they have different types of wavelength of which they absorb light. So that color will activate the light, and then the LED light will.
That's not my question. That's not my question. My question is, say for instance, Helicobacter pylori. It is within our stomach. Okay. So in that case, how will you apply the phototherapy? Will you apply the light?
Currently, it's used. We have a laser technique that we have been using. I get to your question now. A lot of laser techniques have been using this thing that they provide us with.
It is within an instrument or within a room.
Yeah. We have a pen developed technology of light, of which we can just highlight a part of our skin and that image of the skin, and that pen has served as a source of light. So now I get your question.
Okay, thank you so much. Thank you.
Hello, Ashiru.
Yes.
I have only one question. In your presentation, you have mentioned the antimicrobial photodynamic therapy of the curcumin-loaded nanoparticle, but you have not mentioned that how nanoformulation of the curcumin to improve its antimicrobial property. So can you?
I try. Okay, thank you, sir. I try as much as possible to stick to the topic. If we are talking about nanoformulation, it's a different topic. In my topic, I just stated it because a question will rise. Curcumin has one of the most low bioavailability, being it's more available in our water media, that is the aqua media. So right now, the available formulation of curcumin, if it is be used as a novel, it has to be synthesized nano to increase its bioavailability, to increase its side of action as well as its therapeutic action. So the nano formulations are various nano formulations of curcumin have been developed. So right now we are speaking development of it are increased. So my is antimicrobial photodynamic of curcumin, but then curcumin has a problem of bioavailability. That's why I included the nano formulations of curcumin, because we are talking as novel, we are talking of technology, we are talking about advancement. So we can't just talk about curcumin. That novelty of curcumin has to be mentioned. The nano formulation has to come in question. But if we are talking about the bioavailability of that's another topic of its own, I think, and the various method of nano formulations are there for curcumin, the production of curcumin.
Okay, thank you.
Thank you.
Thank you. Now I would like to request Ibrahim Aminu, our today's last presenter. Ibrahim, please start. Ibrahim, you here? Ibrahim Aminu? Ibrahim, you are in the meeting? Anita, sir.
Yeah.
Judges, there is a request. Please wait for some time. Ibrahim will join us soon.
Hello.
Yes, Ibrahim.
Okay. Am I audible?
Yes, you are audible. Please start. Share your slides.
Hello, Ibrahim. First turn on your camera and then share your PPT. Are you able to hear me, Ibrahim? Ibrahim, please reply. Hello. Yes, Ibrahim. Please turn on your camera.
Okay. Thank you everyone for your kind presence, respected judges and dear
Yeah,
delegates.
thank you, Soumya.
Thank you, ma'am. Now, at the end, in the last but not the least, we are sharing a PPT, PPT video of IPGA. Actually, this is a just welcome speech for our 25th September Pharmacist Day celebration. It is provided by Dr. Vijay Bhalla sir.
Welcome to all my fellow pharmacists. I am Dr. Vijay Bhalla, Principal, SGT College of Pharmacy, SGT University. Very soon we shall be celebrating Pharmacist Day, that is on September 25th. World Pharmacist Day was the brainchild of our International Pharmaceutical Federation, that is FIP, to bring attention to pharmacies and the positive benefits they offer when it comes to health. Transforming global health is the theme of this year, World Pharmacist Day. This is the most appropriate theme as we all are facing the challenges of the COVID-19 pandemic. Access to safe, effective, quality and affordable medicines, health technologies and pharmaceutical care services are the major contribution of pharmacy. We are all individually playing a part in transforming the global health of our communities. As pharmacists all over the world are working tirelessly to stop the spread of coronavirus, that is COVID-19, and to ensure people continue to have access to medicine they need. Each pharmacist has an opportunity to communicate how they are transforming health through a variety of health services in their communities, including advising on healthy living, immunoboosting, vaccinating to prevent diseases, and ensuring that medicines are taken correctly, thereby managing disease well and improving the overall quality of life. Pharmaceutical scientists transform and prolong the people's lives by developing safe and effective medicines and vaccines. Pharmacy teachers are also transforming outcomes by ensuring that there are enough qualified and competent pharmacists and scientists to meet the growing need of the society. Indian Pharmacy Graduate Association and Student Forum of IPGA has ambitious plans to highlight the value of pharmacy profession to their stakeholders during the celebration of World Pharmacist Day on September 25th, 2020. I wish them a good luck. Happy Pharmacist Day. Thank you.
Thank you, sir, for your nice presentation. Now with this, I am closing today's first day session of AICT sponsored e-conference on antibiotic resistance, causes and future challenges in digital India. We will see you tomorrow at 10:30 a.m. for our second day conference. 10 a.m. Till then, stay home, stay safe. See you tomorrow.